This year, GYSS celebrates its 10th anniversary of the annual meeting between young scientists and engineers from all over the world, and outstanding scientists and engineers who got Nobel Prize, field medals, Millennium Technology Prizes, and Turing Awards. Last year, besides the problem of COVID-19 pandemic, I had a severe accident, and so I could not join the event even online. This year I can be with you online, and I am allowed to send more Thai young scientists to the event than the previous years. Besides having the opportunity to learn from global scientists and engineers from other young scientists and the visits to Research Institute in Singapore, these young people who are mostly in science and technology careers can be inspired to create the works that benefit Thailand and the world. As they become successful professionals in science and technology, they will inspire other children to pursue the same careers, and the government could see the importance of promoting more citizens to study science and technology. I hope that young scientists and researchers in science and technology won't be discouraged by the obstacles they encounter and continue to do their best. I hope that they will always think of new things as GYSS moves on to the next 10 years.
It's 10 years of Global Young Scientists Summit, and it's my pleasure to congratulate Singapore's National Research Foundation and also the organizers for having GISS this year, and I wish you a wonderful meeting. I'm very glad when I'm thinking of all the participants being the current or the former ones, because having exchange about science is one of the best things you can do. We in Lindau also had an anniversary last year, and of course it's always great to have this recognition for the past, but at the same time it gives much motivation and energy to carry on, particularly in times like these. Thinking of the young people, my message for you young scientists taking up a career in science and technology would be: do what you're really passionate about, be always curious and ready to share your knowledge, and a platform like the Global Young Scientists Summit is great for doing that. Sometimes I'm asked if I could get advice. I'm not within science, but my advice for further careers, of course with reference to a Nobel Prize, would be: despite all very obvious value in applied sciences, let's take COVID-19, do not lose sight of basic research, also for the sake of future generations. And now I hope you enjoy the GISS 22 and wish you all the best for it. Many greetings from Lindau.
The success of the Global Young Scientists Summit is a testament to the importance of borderless collaboration in the pursuit of excellence and the benefit of humanity. As we seek to tackle global challenges such as climate change, biodiversity loss, and protecting human health, and to pursue the knowledge of ourselves and the universe around us, this is the bedrock of future research, and it's your generation that will be at the forefront. The scale of these challenges may have inspired you all. I'm sure the desire to expand our understanding and make the world a better place will continue to inspire your generation. While the challenges are great, your generation, like those before you, stand on the shoulders of giants. You have access to new knowledge and tools that can help change the world for the better. I urge you all to grasp the opportunities and pursue your passions.
I have the great pleasure of introducing Professor Yonath, who was awarded the Nobel Prize in Chemistry in 2009 for her contributions to unraveling the structure of the ribosome. Professor Yonath's impressive career spans more than 5 decades. After receiving her PhD in chemistry, studying X-ray crystallography from the Weizmann Institute in Israel, she carried out postdocs at Carnegie Mellon, MIT, and the University of Chicago in the USA. In the 1970s, she established the first structural biology laboratory in Israel at Weizmann, where she pioneered work on protein crystallography. From there, she went to head the Max Planck Research Unit for Ribosome Structure in Germany. At the moment, she is the director of the Kindelman Center for Biomolecular Structures and a professor at Weizmann. Her current work focuses on protein biosynthesis, the study of antibiotics that hamper protein biosynthesis, on human diseases associated with ribosomal mutations, as well as on the origin of life. She holds an impressive list of awards and achievements, including honorary doctorates from over 45 universities worldwide. Today, she will talk about the design of next-generation therapeutics. And while she couldn't be here in Singapore today to visit us in person, we're very much looking forward to her recorded seminar. And she will be available live after it to answer your questions, so stay tuned to talk to her then.
Good day. I would have liked to be here today with you, but since I cannot, at least I sent my lecture, and I would like to tell you about what is it. It's about the translation of the genetic code into its products, the proteins, and how one can stop it if one wants to stop the life of a pathogen by antibiotics. Antibiotics are fantastic, but there are problems with them. The main problem is resistance, and we are trying now to fight against it, and that's about what the lecture talks. Thank you. I want to talk about next generation antibiotics, or at least what we suggest should be next generation antibiotics. And for this, I also want to show my motivation, which was actually curiosity, like that of our hero, Mr. Einstein, that said that all what he had was curiosity and passion to it. When I became a young scientist, I thought the translation of the instructions embedded in the genetic code into proteins is one of the most interesting in this interesting life process. Therefore, I focused on it. What do I mean? Let's first see what proteins do. And what I use here is some pictures from a children's book about proteins in human. But actually, proteins perform nearly all the essential functions in every living cell, even in bacteria, in flowers, in trees, and in human. So here there are some proteins. Hair and skin, structural proteins, signaling proteins like insulin, transport proteins like hemoglobin, sensory proteins like vision, and enzymes that are taking apart the proteins that we eat to their components in order to make new proteins, those that we need. So what we know about proteins, some of them perform almost all cellular functions, as you saw previously, even muscle motion, and some are responding to pressure, like heat or cold shock, sometimes heart events, and so on. Proteins are long chains made of amino acids. There are 20 types of amino acids. The fold of each protein, which is carefully designed for fulfilling its function, is determined by the sequence of the amino acids that are building it. So what I want to tell is in nature, structure means function. Look at this machine, very sophisticated machine that is called paperclip. It works only when the fold is correct, where the structure is correct. If it's being modified by fingers, without heating, without cooling, without cutting, without anything, just changing this structure or the fold, there is no function. And this is the way proteins work. So as I said earlier, there are 20 types of amino acids, and their sequence within the proteins, which enable its functionality, is dictated by the gene that codes for proteins. So proteins are made according to their genes, which are in the DNA. Actually, the DNA is a string of instructions written in a 4-letter language in which each 3 letters, or we call them also bases, are coding for a specific amino acid. These are called codons, these 3 letters, these triplets. So this is a typical enzyme. It's made of many atoms, many amino acids, each made of atoms. It has a active site. And if we look into the active site, this is here, and this is supported by the structure around it. I rotated it and I enlarged it, and you can see the active site now with the substrate. And you see that the substrate fills it exactly. If it was too large, it won't fit. If it was too small, it will flip around. So it must fit this exact. So this must be exactly according to what nature does. In this case, what nature needs is this for this substrate. So how do the proteins, are they made? Here there are 5 amino acids, 5 components. All of them have, it's part of the 20, yeah? All of them have the same backbone, and all of them have the backbone that can fit each other, and each of them has a different side chain that gives them the biological or the chemical character, and some of the side chains can also be biological tools. They get together, they become more and more and more together, so long that I didn't even draw the details. Then they become long protein. About each normal proteins are about between 200 to 500 amino acids, and it's worth nothing unless it is folded with an active site, as you could see earlier in the real protein. So this was known when I started my work, but it was not known how it happens. This is what I wanted to find out, how this happens. So many other people wanted to do it. They were more experienced and they had better labs, and they failed. So there was a big skepticism about the possibility to understand it. There were even papers saying it cannot be done. Yet I tried to do this. I will tell you afterwards why. I knew that we also can fail, but I knew that if we don't fail, if we make it, we will have a big smiley. And this is what we have today. So the central dogma says the DNA that you can see here is where the bases are. It means the letters, the triplets that code for the proteins. The information is inside here. These are the bases. These are the letters within the double helical structure. And here it is not available for reading. It's covered, it's hidden. So the first thing that happens, there is a transcription, to a very similar molecule that can exist as a single chain. So here the letters are exposed and can be used. They are available for using. And this can be translated to growing proteins by a particle within every cell that is called ribosomes. Actually, there are many ribosomes within every living cell. So they are universal. It means they function in a very similar way in all cells, regardless of the source. It can be bacteria, it can be flower, it can be cockroach, it can be human. They do the same, the decoding and the production of the protein the same way. There is a huge number of ribosomes that function in every living cell. Even in bacteria, they can reach 100,000 when they work. And in some mammalian cells can contain millions. For instance, the liver may have 5 to 6 million ribosomes. In vivo, the ribosomes act continuously. They can form up to 40 peptide bonds in a second. They hardly make mistake. So I, as a very good and fast student, needed 6 hours for one bond, but they can do 40 in a second with about mistake rate of about 1 to a million. So now that we know how they work, we like to describe them as a factory that we call ribosomes that is made of 2 floors. In the top floor, the information is being decoded. The triplets are coming in. It means the whole messenger RNA comes in, and triplets, when a triplets come here, it's found what it is coding for. The amino acid that is cognate to it will come inside into the factory and be connected to the growing protein that goes out. This arrow had to be inside, but I don't know how to draw it, so I drew it outside, but it happens in the factory. Then this track will become empty and join the other empty tracks. And as I told you, there are 2 floors. Here the decoding is being made, and here the making of the protein, the making of the bond, which is called peptide bond. So actually what you saw in the factory is shown here now in molecular way. Ribosomes, all ribosomes are made of 2 subunits, small and large. These 2 subunits are independent in the cell. They live as small and large. They come together when ribosome has to work, and they are connected by contacts between them and by the track, the tRNA molecule that brought the amino acids. So here it is, and it connects between the decoding center and the peptide bond formation center, which is called PTC. So this is what we found after 20 years minus 2 weeks of work, and we made a little movie that is available in YouTube that you can see here. Here, messenger RNA comes to the small subunit that waits. There are factors in the cell that are coding that are telling the ribosomes that has to start to work. There are initiation factors. In bacteria, there are 3 of them, and what you see here is actually ribosome from bacteria. And the whole thing is monitored by them. So yes, there are many more factors. You will see some in a minute. So messenger is reaching the small subunit that takes it. We made the movie, us, together with 2 students of the Art Academy in Jerusalem. And the messenger now will go into its path in the small subunits, the top floor of the factory. And it's monitored by initiation factor 3. When this is okay, initiation factor 2 is bringing it the first track, the tRNA molecule with the amino acids, this one. And now where everything is okay, the factors go out and the large subunit can come and associate with the small one by their surface and by making bridges. Now the ribosome is ready to work. We look at it from the solvent side. Up is the large subunit, below is the small one. And you can see elongation factors bringing it inside the tracks. The tracks are being decoded and peptide bond is being made. And you can see the ribosome helps. And it makes very fast, 40 of them in a second. So you can see here how the decoding happens in the small subunit. And from the large subunit, we left only the tunnel through which the protein goes out. So this is how it is being protected when it's made. Now you can see the whole ribosome again. The tunnel is inside there in the large subunit. And the process continues until the protein is large enough. Here it is being grown and coming out. And when there is a stop codon, a codon that says stop, then instead of tRNA comes a factor, recycling factor and release factor. These factors separate between the small and the large subunit. The protein can fold and come out and has to do what it has. And the tRNAs, the tracks, and the messenger and the ribosome are waiting for the next job. That's all. That's the whole story. So what we saw in the movie was rather fuzzy, but what actually happens is that we know the position of each and every atom in the ribosome, small subunit, which sediments as 30S and large subunit is 50S. You can see that the ribosome is made of many components. The main one is ribosomal RNA, another RNA molecule, and many ribosomal proteins in many colors here. And just to remind you, the small subunit is doing the decoding, and the large is peptide bond, is for peptide bond formation. Now let's look at these people. All of them are giants, either in music, like Mozart, Chopin, Schubert, or very intellectual writers. And what's common to all of them is they died very young. You can see their lifespan. The oldest is 47. Also, what's common to all of them, they died before or at the middle of last century. What you cannot see in the picture, but I can tell you, is they all died from infectious diseases that people don't die now from them. So what happened in the middle of last century? In the middle of last century, antibiotics came into use. And they saved the lives of many, many people and, of course, extending lifespan. So because of the fundamental role played by ribosomes, many antibiotics target them. Almost half, over 40% of the clinically useful antibiotics target protein biosynthesis, mostly by paralyzing the ribosome. What are the natural antibiotics? They are the weapons that bacteria from one type use to interfere with the life of other species of microorganisms when they have fights for space or for food or and so on. Question is, how do the tiny antibiotic paralyze the giant ribosome? So bacterial ribosome have a molecular weight of 2.5 million Dalton and antibiotics less than 1,000. And the trick is that they are bound to the active sites. So you see here the small subunit and the large subunit. And the circles are showing where are families of antibiotics. It means the natural antibiotics that were modified by companies. And you see all of them are bound to functional sites, either to the decoding, or to the bonding, or to the tunneling, or to the hinging. These are the motions that are needed for continuing. So this is the trick, and we want to show you a movie that was made based on the previous movie. And it's already 13 years ago, and as I told you, in YouTube, and it shows that the tricks played by the antibiotics. So this is a small one, edein, that doesn't let the messenger bind. That's all. Second one that you will see is tetracycline. It also works at the small subunit. It doesn't let the second tRNA bind to the second position. So one is bound, but the second cannot. Second position is called the A site, for those that want to know. So just occupies it. Then I want to show two that work on the large subunit. First is erythromycin, which is one of the first that came into use in medicine right after penicillin. It sits in the tunnel. If you remember, the tunnel that protects the protein and doesn't let more than a very short protein to be made, and then it blocks. And the last one that you will see is disturbing the formation of the bond, very clever chemically, maybe the most clever chemically. So here the bond is being made, and it just sits in the bond. No bond, no more protein. That's the whole story. So, all ribosomal antibiotics bind to ribosomal functional sites. We just saw it. But these are highly conserved because all ribosomes work the same way. I already said it. And mandatory for clinical use is the distinction between the patient and the pathogen. So how does this happen? This happens by subtle differences. Even in highly conserved regions, there are subtle differences. And I want to show you one to explain. So if we look into it, we look into the whole large subunit. This is the entrance to the tunnel, and erythromycin blocks most of it, enough not to let proteins go through it. So if we look at this position in the tunnel, like a perpendicular to its long axis, we see that there is a pocket, and the pocket is made just from ribosomal RNA, and all the members of the erythromycin family, which is called macrolides, are bound here. So some are bound excellently, some are bound very good, some are bound just good, but all of them block the tunnel. And all of them interact with one of the components of the tunnel that I marked as a pink, and it's adenine A in position 2058, according to E. coli. So this is correct for all bacteria. All eubacteria have A in this position. Let me show you how it binds erythromycin. So this is this A, and this is erythromycin, and affinity here, the chemical affinity is very, very good. It means this is very cleverly designed by bacteria. Now, please, ladies and gentlemen, pay attention to this point, and you will see the difference between bacteria and us. This is all bacteria and us. But when there is a G here, us, there is repulsion. This is too close here, the distance, and there is no binding. That's all between bacteria and us, between bacteria and any higher organism. Second question that I want to discuss, second point, is resistance. Resistance to antibiotics is one of the most severe problems in modern medicine. And let's talk about it. First of all, how it is being acquired. So I will show you one of the methods. Selectivity was shown here between this, and the bacteria can just change themselves to G, and now they are resistant. Actually, resistance to antibiotics is a basic process for the survival of many microorganisms, regardless of their exposure to modern clinical treatment. And this is a very important finding. And this is one of the most severe problems, as I said before. The World Health Organization said we are reaching post-antibiotic era. It means the time that antibiotics will not be useful anymore. And even the World Bank estimated that up to 3.8, almost 4% of the global economy will be lost by 2050 because of resistance to antibiotics. Can we combat antibiotics fully? In my opinion, not. Because bacteria want to live. Maybe want is a street word for it, but this is what happens. And they are cleverer than us in terms of survival. But we can do some things. So our lab decided in our lab to help it, because companies are hardly interested now in antibiotics. Most of the big companies are not interested at all. So until recently, all the known structures were of models for pathogens, not real pathogens. And this explained the common modes of antibiotic action, as I showed previously, but they do not explain specificity of pathogens. And this is what we wanted to see. What's the difference between non-pathogenic and pathogenic? So we crystallized the large ribosomal subunit from a pathogen, actually a multi-resistant pathogen called Staph aureus. You can see here the crystals. And we got the structure. I won't show the whole structure. I will just show some lessons we studied by comparing it to non-pathogenic. So first of all, it's possible to increase antibiotic activity. More potent antibiotic can be made. We collaborated with a company called Nabriva, a small company in Vienna, and they made 16-fold stronger antibiotic. Lesson number 2 and 3 I skipped. Lesson number 4 is beyond my expectation. Novel antibiotic binding sites. I didn't expect it. It's a very, very nice surprise, and in my opinion, also very powerful. So if we look here in the structure of the large subunit in gray, this is the skeleton in two views. This little bluish, this little cyan, show where there are additions on the skeleton of the large subunit. You can see some on this side and some on that side. And maybe you can see even better here from all sides. We drew here only those that are on the surface, and those that exist only in Staph aureus, not in the pathogen models. So we suggest to use them as new positions for potential binding site, antibiotics potential binding site. And I want to show an example here. This is part of the ribosome. There are here two structures, one of the model and one of the pathogen. Here they are indistinguishable, but here there is a little addition. This is what you saw in this view. And we suggest that this can be potential binding sites because it's only in the pathogenic, and we found that it is also important for protein biosynthesis. So actually in a model system, we identified 25 new potential binding sites, unique, and 16 of them where they were blocked chemically, they inhibit protein biosynthesis. And because these sites are not involved in the primary ribosome activity, namely in the decoding and peptide bond formation, no pathogen contains genes for their modification. It means the bacteria did not understand now, although they are clever, did not understand now that they are important. So for now, they can be exploited for the design of specific advanced degradable antibiotics, hence environmental friendly. So I talked a little about specificity, I'll talk about it a little bit more, and about degradability. It means environmental concerns. So first, specificity. I hope that you all know about the microbiome. The microbiome is where trillions of bacteria reside in our body. They are called the good bacteria. They are usually in semi-exposed organs like guts, ears, lung, and skin, and they are mainly harmless. But they are susceptible to antibiotics. And so if antibiotics can penetrate into where they are, they can kill them too, and then people can catch other diseases. Therefore, we suggest to exploit these sites and design those that will not disturb the microbiome, the good. Also, there can be degradable. What do I mean in degradable? This is the environmental and ecology consideration. This is based on the fact that most of the ribosomal antibiotics that are used today are extensions of small organic inside the cores that are not digestible, and when they go out from the body with the rest, with the sewage, they are not degradable, and therefore they can penetrate into the water. They are too small to be used, to be found by most of the used facilities for purification, and then they go into the water. They can penetrate into the irrigation water, and they come back to us. So this is why when we design our antibiotics, we want to make them degradable, biodegradable. So now I want a few words. How did we start? Because I thought that it has some lessons that we can learn from it. First of all, why did we use X-ray crystallography? Because human can see, I think, maybe insects with normal microscopes, cells, bacteria, viruses, but not atomic detail. For this, we need crystallography. For crystallography, we need crystals. They diffract. We see the diffraction here. They diffract according to the Fourier transform, and if we collect the whole three-dimensional space around the crystal, we can get enough information to build back the structure. So here you can see X-ray coming. There is here a crystal. We collect the data with computers and with knowing how to do it. This little thing is the crystallographer. It means like something like me. And then we get the structure. So we can get structures of many types of molecules. But we need crystals. Let's define what crystals are. So this is sodium chloride. It's only 2 atoms, sodium and chloride. And it likes to make a 3-dimensional periodic system, even in nature, spontaneously. So you can see full periodicity in 3 dimensions of chlorine and sodium in between. This is a natural crystal. This is a crystal of a protein that was first crystallized in Oxford in the 60s, but a 6 years grader, about 12 years old girl, could repeat it. So proteins can be crystallized by this very complicated, now we know, very complicated structure that you know. But even before the structure was known, there was a big question mark. Actually, this question mark said no. And the people that tried to crystallize it, they explain it by all types of high degree of internal mobility, flexibility, structural heterogeneity, chemical complexity, all correct. But they paid attention less to a very important property of ribosomes, that they have marked tendency to deteriorate. Even when we prepared it, they deteriorate, and we get less than half of the expected activity. And I was much more open to this problem. So I thought, first of all, we have to find ways that the ribosomes won't be deteriorated when we made them. Okay, I thought about it, but I didn't know what to do until I read a paper about is the metabolism of the sleeping bears in the North Pole. The North Pole, the bears are going to sleep for the winter, and a delegation went to see what happened inside them. So they found out that in every cell, inside the membrane of each cell, the ribosomes are fully organized periodically. Monolayers, not real crystals, but monolayers of organization, and this shows that ribosomes can be orderly packed. Every bear, every cell. So then I thought, why is it? Why did nature provide this mechanism? And in my opinion, this is the way to maintain a pool of active ribosomes for the end of the winter, when the bears would like to do other things and they need proteins to do them. So my conclusion was that stress induces periodic packing of ribosomal particles. So I thought, let's look at stress, at bacteria that grow under stressful conditions. I didn't want to work on the bears, on the ribosomes of the bears. First, I didn't want to kill more bears, and more important, because the ribosomes from bacteria can be useful for medicine, whereas those of bears not. So the solution was to use ribosomes from bacteria that grow under stress, like in the Dead Sea, in hot springs, or in waste of nuclear facilities. I just want to show you the Dead Sea. It's the lowest point in the world. It's in Israel. You see, from top, no animal, no tree, no nothing, no life. You can see from closer, these are all structures of salt that came out by evaporation. No animal, no plant. But there are two types of bacteria, and I want to show you the colonies of one of them, Heloarcula maris morti here. You can see it here too. And I knew about this bacteria, and we used its ribosomes. Also, we use ribosomes of Deinococcus radiodurans, which is a bacteria that can grow under super extreme conditions, very hot, very cold, environment, dust, no food, even under irradiation. And we got beautiful crystals. Look how nice. So we got actually 25 types of them. I show here only 6, those that were useful for crystallography. And I want to show you 6 years after we started, we went to measure them. We took crystals to measure in this accelerator, which is in Grenoble. In Israel is a member of it. Actually, we used another one, but it's such a beautiful picture, I decided to use this. From top, it looks like that. Look how wonderful. From getting close, you see how it works. So here there are particles that be an accelerator, then they run around in this ring. And in the tangential, we have stations. Here there is a station that Frank is measuring on ribosomes. So each station has its own properties. Some are useful for ribosomes. So in 1986, 6 years after we started, we got the first diffraction. Look how beautiful, beautiful spots all the way to the end from the last subunit of Heloarcula in time equals 0. We were really very happy, but not for long. Second picture was already deteriorated, damaged. It means the X-rays damaged the crystals. We repeated it 100 times, and we didn't get anything better. And we felt like we started from here, and we saw only this mountain. We call it the Everest, sorry. And we climbed it, and climbed it, and climbed it. Took 6 years until we got to the summit, and what did we see then? Not a really big, much bigger Everest, much taller Everest behind, that we will have to get there in order to measure. Much more difficult than until then. So this was the time I thought maybe we should stop this project, but instead I thought about why does it happen? All living tissues suffer from X-rays, but this is much too much. So I explained it by the crystals being weak or very soft, and the damage occurs in 2 steps. The primary is local disruption of the bond, and then when the bonds are being cut by the X-ray, the 2 parts of the bond are unhappy, going around and break more bonds in order to satisfy themselves, either to give an electron or to take an electron. And we thought that maybe the way to stop it was to cool the crystals so cold that there will not be energy for the running around. And this worked, although everybody didn't think so. Even I, look, this is the experiment done at Stanford. Look at my face. Even I didn't believe in this, in our own experiment, but it worked beautifully. Have a look. And 20 years later, look how happy I am. Now it works. And this is the machine that cools, and this is the machine that measures the X-rays. This is in Grenoble. Actually, not only for us, it worked for the whole world. We introduced it here at the end of '86. Within the beginning of '87, 90% of the world used it, and now everybody. So what you see here is the number of structures that are known now and have been deposited in the protein data bank, and this is the year. And have a look, this is only till 2011. 47,000 structures, all of them done, or 99% of them done in our cryo method. And all of them are of very important materials for medicine or for making new materials. Actually, now it's 217, and there are 80,000 new structures that couldn't be done otherwise. So how about future? We go back to the lecture, and what I want to tell you, we plan to identify the specific structural features which are unique for each pathogenic bacteria. What I showed you about Staph aureus, we want to do for all. Once we identify such features, we will design novel antibiotics for them. In these studies, in the novel antibiotics, we shall use cryo-electron microscopy, which provides high-resolution structures with no need of crystals. All what I told you until now about crystals, we will not need them in the future, taking advantage of the current revolution in biostructure research. This is our program, and I hope that you support us. So before I finish, I want to thank the Weizmann Institute that let me work. Here is the president that made the decision, Michael Sella. He was a biologist. Although I was called the dreamer of the world, he still supported me. And when he finished his term, he was replaced by Chaim Harari, who is a physicist, and he also supported me. They were encouraged by the Scientific Advisory Committee, about whom I want to highlight Alex Rich, who tried to crystallize ribosomes with no success for 10 years, and he was still very supportive. Actually, the whole work started with a very strong collaboration with Max Planck in Berlin, with Dr. Wittmann, that he really wanted to see the structure of the ribosomes, and he helped in establishing a group for us in Hamburg, near the synchrotron there. He died 10 years before the structures came, unfortunately, and then he was replaced by Franceschi and then by Fucini. So I want to thank my research group in the two places for their devotion and determination, in good and in bad times, for many years. Here, this is the group in Hamburg. It was terminated in 2004. It always had angels. The technical assistants say that otherwise they cannot work if they are not angels. Ribosomes are too complicated. And the group came to the Dead Sea to look for new bacteria. Well, they had good time. The group in Israel is still working. Here you can see people that came and went through the last 3 years. Some of them are still with us. I want to highlight Dr. Anat Bashan, who is the senior scientist that runs the show all the time when I'm doing other things. And I cannot show you one by one, but I want that you pay attention to Tamar. Tamar came 16 years ago for 10 weeks. She's still around. At the day that I took a picture, she had birthday, so she baked a cake. You want to see the cake? Here is the cake. And it shows that in my group, ribosomes are considered sweet. I also want to thank my family. I cannot show you all of them. My mother, my sister, and so on. Just my daughter, who is a doctor, a medical doctor, and her daughter. This is the girl that crystallized the ribosome when she was 12. And they were also supportive all the time, even when I wasn't there. My granddaughter actually gave me a speech and then a prize. I will show you the prize in a minute. In the speech, she said, as you know, she is a very busy scientist, but she always finds time for me. And this is the prize that she gave me. No, not this one. This is the medal, Nobel medal. Her prize is. The grandma of the year is Ada Yonath, at least as important. And when I asked her, what is the year? every prize has a year. She said, you have to reprove yourself every year. I'll take it off the wall if you fail. Still on the wall, you see? So ribosomes became popular. In a carnival, he is me. And she, his wife, is a ribosome. Look, protein comes out of her. Also, younger children became ribosomes. And this is what happened to me by one of the best artists and writers in Israel, Michel Kishka. And if you have a look very carefully, this is the large subunit, and this is the small one, and it's symmetrical. And he thinks that the best place for antibiotics is here and here. Thank you. Okay, I hope that you liked it, and I hope that something will come out of it, at least mentally, intellectually. And as I said, I would have liked to be here with you today, but since I cannot, good day to all of you.
Thank you, Prof. Yonath, for the really inspiring and impressive talk. I know, we learned a lot from your lecture and have a lot of excitement from a lot of the students here. We have a ton of questions that we're hoping that you can answer for us today. So I'm going to start with the first one from Nuke Patal. They say, thank you, Prof. Ada, for a great talk. What do you think is more powerful, either targeting the ribosome function or targeting the tRNA synthetases, as both are crucial in protein translation?
Both are okay, as long as it works. If on the ribosome, we try to find specific structural motifs that are different between the pathogens and the non-pathogenic, the microbiome. So we can distinguish between them. But there are very few of them, and it doesn't work very smoothly. There are problems with this too, but the idea is to target only, or to avoid the protein synthesis only in the pathogens. If you can do it also with the synthetase, or better you can do with the synthetase, it's okay. It doesn't have to be ribosomes. It just happened to be a good target, and we can find specific motifs, structural motifs.
Okay.
But if you come up with another system, go on.
That's great to know. All right, we're going to move to the next question from Miro. They're saying that they are a physicist doing molecular dynamics, and they're wondering which biomolecular system you think is most critical to understand the dynamics. Is it the ribosomes?
Dynamics of what?
Dynamics of
dynamics of peptide bond of forming proteins. This is the best to look at, but depends which dynamics.
Yes.
This is also dynamics.
Very true. Okay, we have a question from Igor. They ask if you think that advances in computational methods, such as machine or deep learning, can be used to predict how bacteria become resistant to antibiotics and therefore be used to combat it.
I think in principle, it's a very good idea. But in practice, for the ribosomal antibiotics, half of the antibiotics go to the ribosome. The other half go anywhere else. Those that target ribosomes, we understand how they become resistant by now. We worked on this 15 years, 16 years after we had the structures. We collaborated with some very good laboratories. We did also ourselves. And this we understand. Now we have to find out how to kill it, how to stop it. Stop the resistance or bypass the resistance. So we are doing bypassing. But also we have now a new project, how to stop the resistance by changing the antibiotics. So far it didn't work. Not only for us, it didn't work at all, except for one case. But whatever works, it's the problem with resistance to antibiotics is so serious that we don't choose whatever we can get or anybody can get.
Yeah, that's a great perspective. Okay, we have a question from Kasinan. They're saying thank you very much. They have a science fiction question. Is it possible that mRNA from the COVID-19 vaccine can be translated by a human ribosome?
Yeah, in principle it is, and it actually, that's the way the vaccine works. The vaccine is pieces of mRNA that code for the virus proteins. In this particular case, it's the how you call it, the chip that goes into the cells. And it is being translated by the human ribosomes. That's the way the vaccine works.
Yeah.
Then the protein is being made, the protein of the virus is being made just by this piece that is being translated, and it evokes, because it's not human, it evokes a human immunology against it, and that's the way it works. I think it's a fantastic idea.
Yeah, it was a great advancement.
What do you say?
It was a great advancement, that's true.
Yeah, also from the point of view of idea, not only from the need.
Yeah. Okay, we have another question from Nuke Pakal. They ask that now that cryoEM has been revolutionized to how we study proteins, what is the most important limitations of this technique compared to X-ray crystallography that you pioneered?
Yeah. Okay, I also worked on cryos, so I pioneered this too. The cryo-electron microscopy works very efficiently and very cleverly on molecules that are large enough to be seen by it. It means we tried to look for every ribosomal protein alone. Cryo-electron microscopy did not care for it, only in the ribosome, because ribosome is large enough. So the advantages of cryo-electron microscopy is that one doesn't need crystals. It means one can look at several states, several functional states, and one doesn't have to crystallize. And crystallization is not always easy, not always automatic, and it freezes the structure, the ribosome or the protein, in a specific conformation. And in cryo-electron microscopy, if it goes from this to this to this to that, and they all exist together and there are enough of them, and the person is trained to look for different conformational states, it can be seen. So dynamics of reactions can be caught. But the best of it is the speed. The first structure came out of our lab, we were the first 20 years. Second one was 6 years. Then we had some structures of 3 or 4 years. And now we have a structure in a few weeks. It's true that it's all built on the first structure. It means we have coordinates, we have already in our mind how it can be seen. And then if it has dynamics, we see the dynamics of it. So this is true, but the time, the facts that no crystals are needed. Crystals are freezing the structure in a fantastic, specific conformation. So we are not frozen in a conformation. We can look at many conformations. This is a big advantage.
Great, thank you so much. Okay, we have a question from Remy. They say, thank you for the lecture. If the active sites of antibiotics bind, the active sites that antibiotics bind to are highly conserved, then why is antibiotic resistance on the rise? Are they silent mutations that prevent the binding, or is there another mechanism that's happening here?
So I think that I showed one, as far as I remember, one mechanism. But the idea of the mechanism is the antibiotic resistance is a result of a change, either in the structure of what hosts the antibiotic or in the structure of the path to the hosting place, a change that will not allow the binding or will reduce the binding contacts so much that it will still be a binding of antibiotics, but in a rate that is not serious, does not cause diseases or cause very limited diseases. Mm-hmm. But anyway, we have to get rid of it.
Yeah. Okay, great. So Vrinda is asking, what do you think will be more powerful going forward to help speed up the drug design? Would it be designing new drugs for current drug targets or finding new drug targets, which then can allow us to potentially reuse old drugs?
I think both, especially if the first steps are by computing. So one can compute both. We looked at the ribosome so much that we decided just to look for specific functional entities that do not exist in the non-pathogenic, that exist only in the pathogenic. But it's not. It doesn't say that all the antibiotics have to be like this. Mm-hmm. For the ribosome, I think also because the good bacteria and the pathogens are sitting together in the body of human or maybe an animal, and the distinction between them is also very important. Mm-hmm. So if you just go to the binding site, and the binding site is more or less the same in both, then if there is a little change, even just one chemical change between the two binding, but this chemical entity is very important, then it's already resistant.
Yep. Great. Okay, we have a question from Belinda. They're asking, when developing biodegradable antibiotics, how do you ensure that the antibiotic can be degraded after use, but not inside the body? And this is in order to maintain bioavailability and avoid things like protease degradation and things like that.
I don't think that there is one answer for this. I think it depends on the compound, on the chemistry of the compound. In our case, our what we call antibiotics, or ours, they are all degradable. And they don't exist in the human body, in the animal, and not in non-pathogenic bacteria. So the non-pathogenic bacteria, the microbiome is not affected.
Mm-hmm. Okay, great. So question from Udi. They say, thank you very much for the important talk. And they ask, so releasing cutting-edge antibiotics might accelerate this resistant formation and lead to super resistant bacteria. So they're asking a question about, should we slow down the race for looking at new antibiotics then?
Because we are afraid of new bacteria?
For new super resistant bacteria is what they're...
Well, I'm not sure it would convince me to stop. Yeah. I think that we suffer enough from the bacteria, the pathogens that are existing. And now there is an epidemic around the world that in what we call normal years, from diseases, simple diseases, there is a lot of deaths or very serious diseases, and we want to get rid of that.
Mm-hmm. Well, we are glad you're continuing the research. Okay, we have a question from Lucia. They say, thank you for a great lecture. Can you please explain the mechanisms of how to increase antibiotic effectiveness other than using a combination of ATB positive and non-ATB pharmaceuticals?
I don't really know what are these pharmaceuticals. I cannot, I'm sorry, I apologize, but I cannot answer this question.
No problem. We have many other questions. We have about 1 minute to go, so maybe I ask this last one to you. Verinda asks, since proteins are so dynamic, do you think the structural information from X-ray crystallography is enough for us to design efficient drugs, or are we missing some information?
Of course, we are always missing something. If we knew everything, the world will be paradise. So we don't know everything, but whatever we can find and progress with it, I think it's a reason to be happy and to welcome it.
Great. Well, thank you so much. You have many more questions. There was a lot of interest. We learned a lot from your talk and appreciate your time. Thank you so much for being here to answer all of the questions.
Thank you for listening.
Thanks.
Thank you very much for listening. For me, it's always challenging to come to GYSS, to your meeting. I come every year.
Yeah. Hopefully next year you can come.
Sorry?
Hopefully next year you can come in person.
Yeah, hopefully too. But whatever, the interaction with young people here, with young scientists, is really fantastic. You see how many questions they have.
Yeah.
So I thank you very much for coming to the lecture, for listening.
Thank you so much.
Okay.
Hello, everyone, and welcome to this final plenary lecture at the 10th Global Young Scientists Summit. My name is Johnson Goh, and I'm from A-Star Singapore, and it's my pleasure to introduce our speaker for tonight. He's Professor Stuart Parkin. Stuart Parkin is a director of the Max Planck Institute for Microstructure Physics in Halle, Germany. He's also an Alexander von Humboldt Professor at the Martin Luther University in Halle-Wittenberg. His research interests include spintronic materials and devices for advanced sensor, memory and logic applications, oxide thin film heterostructures, topological metals, exotic superconductors, and cognitive devices. Parkin's discoveries in spintronics enable a more than 10,000-fold increase in the storage capacity of magnetic disk drives. Now, for his work, which enabled the big data world of today, Parkin was awarded the Millennium Technology Award from the Technology Academy Finland in 2014, and most recently, the King Faisal Prize for Science 2021 for his research into 3 distinct classes of spintronic materials for memories. Parkin is a fellow or member of the Royal Society of London, Royal Academy of Engineering, National Academy of Sciences, National Academy of Engineering, the German National Academy of Science, the Royal Society of Edinburgh, Indian Academy of Sciences, and TWAS Academy of Sciences for developing these spintronic devices, and he has received numerous awards from all around the world. So without further ado, let's welcome Professor Parkin.
Okay. Well, good afternoon. I'm very pleased to be here and delighted to tell you something about my research. And I want to tell you something in particular about a novel technology that we've been working on for some time that could replace many of the technologies used today to store digital data. And this is something I called the magnetic racetrack memory. So I'd like today to tell you something about memory on the racetrack. So if we move to the first foil, let me just point out, this is my current institute in Germany. I moved there 5 years ago from sunny California, and now I'm very happy to be able to take delight in the seasons of the year. This is my institute that I photographed last year in the winter, where in Germany there can be quite a lot of snow. Nevertheless, the Max Planck Institute is a great place to carry out exploratory research. The Max Planck Society is one of the leading research organizations in Europe with more than 20,000 employees and 84 independent institutes, of which this is one. My own institute is interested in developing novel materials that can impact future technologies. So I want to take you back 550 years to a very important technological revolution that took place beginning in Germany by a technologist and engineer, Johannes Gutenberg. He invented what is called the movable type press. Before his work, before his discoveries, information was stored in the form of just written words on pieces of paper. And this meant that it was limited to small numbers of individuals. With his development of the printing press, books could be printed more than a single copy at a time. And this enabled information to spread widely and led for the next several hundred years, if you like, the age of enlightenment, when information could be accessed by people by going to read books in libraries. Well, this era of analog printing lasted 500 years. But 50 years ago, the IBM Corporation in California, in San Jose, invented a new technology for storing not analog data, but digital data in the form of zeros and ones. And the information was encoded in the direction of magnetization of small magnetic bits that were encoded in a thin magnetic film deposited on the surface of a disk. Today, that disk will be made of glass, and the disk is rotated mechanically, and a device is moved over the surface of that disk for reading and writing that magnetic information. So over the last 50 years, the storage capacities of a magnetic disk drive has increased by 1 billion times, and the cost has gone down by even more than that per bit. But the last 4 orders of magnitude improvement were due to a spintronic device that I invented 30 years ago. And I want to tell you a little bit about the physics behind that device, a spin valve, in the next few foils. In this view graph, you can see well, you can't, on the bottom left, you can see a magnetic force microscope image of a series of these bits. The black and white regions correspond to magnetization pointing out of or into the plane of the magnetic media on this disk. And over the years, the objective was to shrink these magnetic regions to smaller and smaller dimensions so that more information could be encoded in a single disk, leading to today's disk drives that have terabyte capacities. Now, the fact that today 70% of all digital information is stored in these magnetic disk drives, even today, and this means that if you look inside what we call the cloud today, you would see millions of disk drives in addition to millions of computing cores. And the fact that this digital information can be accessed by modern day communications is the basis for some of the most valuable corporations today, like Facebook and Google. But moreover, this data is a very important commodity that allows us, for example, to make scientific discoveries more rapidly than was possible before. And this, we could look at, for example, this very nice paper published in Science 10 years ago at how the world's storage capacity has evolved over this 2-decade period of time. And what these authors looked at was how that information was encoded. The orange corresponds to devices that are essentially analog, like books, newspapers, and so on. And the blue corresponds to devices that innately store digital information in the forms of zeros and ones. So shown here, for example, 10 years ago, much information was stored in CD compact discs. That no longer is the case. And if we look today, we would see that 100% or 70% of all digital data is stored in the form of magnetic disk drives, and the rest of the data is stored still in digital form. So we've gone from the analog era of Gutenberg to the digital data of today. We've gone from books in libraries to magnetic disk drives in the cloud so that information is much more readily available than before. And that has enabled us to expand both our scientific and technological frontiers. I want to say a little bit then about what is spintronics and what is this spintronic device. And so I just remind you that today's computing and memory technologies use the electron's charge. In a transistor, a switch, we turn on and off currents of electrons by applying voltages. This is basically the essential computing element of today. On the other hand, we can build memory devices by capacitors in which we store certain numbers of electrons. This would be, for example, a flash memory. But the electron has another property called spin. It's a quantum mechanical property that was discovered 100 years ago, and the spin is of 2 flavors. It points up or it points down. So it's innately digital. And we can, over the last 30 years, we've developed a field of spintronics in which we can create, manipulate, and detect currents of spin-polarized electrons to make novel devices for memory and potentially computing applications. I'm only going to talk here about memory and storage applications. So I want to just give you a brief insight into the device that makes possible the super high storage capacities of magnetic disk drives today. And it's a device that can read tiny magnetic fields. So let's take a metallic conductor like copper. If we introduce a current from a battery, then there will be equal numbers of electrons with spin up and spin down. They equally carry the current. So the current has no net spin polarization. Now, of course, the scattering of these electrons determines the resistance of this material. Now, on the other hand, if we take a ferromagnetic material, this is colored blue to represent a blue direction. When we introduce current from a battery, within a very short length scale, the mean free path, the electrons will be scattered. And the electrons which are polarized with their spin up or polarized with their spin down as compared to the magnetization direction that sets the direction of the electron spin will be scattered at different rates. In this particular material, the spin-up electrons are scattered strongly, while the spin-down electrons are scattered only weakly. And this means that the current in this material is carried by electrons largely of one spin polarization. This, again, was a concept that was developed only shortly after the concept of the spin was evolved. Now let's take this device. In this cartoon, you can see two magnetic layers, a red layer and a blue layer, where the color represents the direction in which the magnetic moments point. And these two magnetic layers are separated by an ultra-thin layer of copper, which is a metallic material. So now when we introduce current from a battery, the red layer will support electrons which have their red polarization polarized up, and the blue layer will support electrons which have their polarization in the opposite direction. And because this is a metallic system, electrons from the red layer will cross this copper layer and then be scattered in the blue layer, and vice versa. So innately, this system where the moments are polarized of the two magnetic layers are antiparallel will have a high resistance. The electrons will always be scattered strongly somewhere in the layered system. On the other hand, if we rotate the two magnetic layers so they're both blue, they both had the same direction of magnetization, now the blue electrons can be weakly scattered everywhere in the system, and this leads to a lower resistance. So we've built now what I call the spin valve. And this spin valve then is a device whose resistance depends on the directions of these two magnetic moments. Low resistance when the moments in the sandwich are parallel, and high resistance when they're antiparallel. And we can then use this to build a magnetic field sensor. If you put the device in magnetic fields, they will cause rotation of the magnetic moments. Let's say if the two moments were antiparallel, then we would change the resistance. And so fundamentally, this is a magnetoresistive device. Now we make a multi-layer of ultra-thin, let's say, cobalt layers separated by copper layers, and we design a structure in which those magnetic moments in the superlattice are anti-parallel to one another in zero magnetic field, as shown in this cartoon. When we apply a magnetic field, then we can cause the magnetic field to rotate all the magnetic moments in the superlattice so they become parallel to one another, and this leads to a drop in resistance. Now, this is called giant magnetoresistance, which was first observed by Albert Fert and Peter Grunberg in superlattices formed from iron and chromium layers made by molecular beam epitaxy, and they observed this phenomenon at very low temperatures and these same very large magnetic fields. Here you can see the field required to change the resistance of this superlattice is 30,000 Ersteds. Although in this case, using cobalt and copper material I discovered, you can see these effects even at room temperature and above, making them technologically useful. However, these fields are 1,000 times or 10,000 times higher than the magnetic fields in a magnetic disk drive, which we would use to detect a magnetic bit. So this is the reason why the spin valve device can be designed to detect tiny magnetic fields. We can make it responsive not to fields of 10,000 Ersteds, but to fields of 1 Ersted. And this is what I did. And one of the reasons I could do this was I demonstrated that in a superlattice or a multilayer, as you change the thickness of the copper layer, as shown here on the, say, on the left-hand side, you can see this plots the magnitude of this giant magnetoresistive phenomenon. In other words, what is the percentage change in resistance of the superlattice when the moments in the superlattice have their neighboring moments antiparallel versus parallel? And in this plots, you can see that the giant magnetoresistive value at room temperature oscillates from large values, for example, in the 100 direction of copper, 60%. When you change the copper layer thickness by just a few angstroms, then the giant magnetoresistance effects disappears. There's no change in resistance. When you increase the copper thickness a little bit more, then you establish the giant magnetoresistance effect again. So the magnitude of this effect oscillates as a function of increasing separation of the magnetic layers here through copper. And this is something that I discovered and was the first to demonstrate. And it also showed, as in this case here, depending upon the crystal orientation of the superlattice, whether 100 or 110 or 111, the period of this oscillation varies. And without going into too many details, the period of the oscillation in the magnitude of the GMR is reflective of the electronic structure of this material. In fact, these periods correspond to wave vectors that span the Fermi surface of copper in those particular directions. And these oscillations are actually reflective of an oscillation in the magnetic coupling between the successive magnetic layers that depends on the thickness of the copper layer. As I said, these are all at room temperature, so it makes these effects super interesting and useful. And just to explain briefly what gives rise to this magnetic long-range oscillatory coupling is a spin polarization of the electron gas in the system. So as shown in this cartoon, if you introduce a magnetic spin into a non-magnetic metallic host, then the electrons will try to screen that magnetic impurity. But because it's magnetic, the response itself is spin polarized. And the spin polarization of that electron gas can then lead to a long-range magnetic coupling. So as shown in the bottom part of this foil, if you imagine two magnetic films shown here as blue and red, they were separated by copper. The spin polarization in the copper has a period of its polarization oscillatory behavior, which is reflective of the electronic structure, cannot be changed. So depending upon the separation of these two magnetic layers, the second magnetic layer will either see a spin polarization of the electron gas, which is in the same direction as the first magnetic layer, or in the opposite direction. And this leads to an oscillatory long-range exchange coupling between these magnetic layers, which is useful for spin engineering interesting structures in superlattices. And again, this is something that I was the first to show and demonstrate. And also, I demonstrated that actually this is the property of nearly all elements in the periodic table, all metallic elements, all the transition metals, whether they're 3D, 4D, or 5D, and all the noble metals, copper, silver, and gold. Each of these materials has a distinctive response to a magnetic impurity or a magnetic layer, which gives rise to this long-range magnetic coupling. And it varies in a highly systematic way as we fill the D bands in these 5D, 4D, and 3D metals. And you can see from this plot of the strength of this long-range magnetic coupling mediated by this spin density wave that the strength is strongest as you increase the D band filling. Now, why is this useful? It turns out it's actually critically important because we can use it to design magnetic structures that would otherwise not be possible. And so if we look on the left side of this cartoon, you can see a schematic of a spin valve structure. The red layer at the top is a magnetic material whose moments are pointing to the right, and then the yellow shown as red and blue, the magnetic poles at the edges of the device give rise to dipole fields. And these dipole fields on nanometer scale are very strong. And this means, that's why I've drawn this device showing the red and blue layers are antiparallel. The dipole fields will automatically align the two magnetic layers antiparallel because of the strength of this dipole field. So we have to find a way to eliminate these dipole fields to make useful devices that can respond to tiny magnetic fields. And again, this is a proposal I made. I said we could build an artificial anti-ferromagnetic layer, as shown on the right, in which the blue layer is replaced with a sandwich of a blue and red layer separated by these pink dots, which is an ultra-thin layer of ruthenium, which gives rise to this long-range oscillatory coupling. But in this case, the thickness of the ruthenium, it turns out, when it's very thin, gives rise to a very strong anti-ferromagnetic coupling. So we can build a reference magnetic layer whose net magnetic moment is 0, but the interface with copper is magnetized. And it turns out I also showed that the phenomenon of giant magnetoresistance is dominated by spin-dependent scattering at the interfaces between copper in this case and the neighboring magnetic layers, which would be cobalt. So by spin engineering and creating an artificial antiferromagnet by building structures with these layers. Each of them is just 1 or 2 atomic layers thick. We can build a structure which is unresponsive to these dipole fields, but then can be responsive to external magnetic fields. And the very first device that was introduced into a magnetic recording head by IBM used exactly this structure that I invented, a synthetic antiferromagnet stabilized by exchange bias with an antiferromagnet, a copper layer, and interface layers designed to optimize the magnitude of the giant magnetoresistance. And basically, we design a structure in which the 2 magnetic layers actually have their moments opposed orthogonally to one another. And when in the presence of the stray magnetic fields, the fringing fields from magnetic bits in the magnetic layer in the magnetic disk drive that are oriented antiparallel, these domain walls will have fringing fields which are detected by this recording head. And this device that I just described can detect these tiny fringing fields, which are 10,000 times smaller than any other device before this device. So Spintronics was able to build, using these concepts of long-range oscillatory coupling, interfacial spin-dependent scattering, the synthetic antiferromagnet, and the proper materials, cobalt and copper, could operate a device at room temperature that was 10,000 times more sensitive to tiny magnetic fields than any other device, and that's still true today. So this enabled then, within just a few years, the area of these magnetic bits to be shrunk by 1,000 times, and thereby to increase the storage capacity of a magnetic disk drive by many orders of magnitude. This was the first success of a Spintronics. Now it turns out the same device where we replace copper by an insulating material turns out to have larger values of magnetoresistance. The physics now has nothing to do with giant magnetoresistance and spin-dependent scattering. It's due to spin-dependent tunneling. So in this cartoon, you can see 2 red layers, 2 magnetic layers with their moments parallel, separated by an ultra-thin insulating layer shown in gray. And this will be approximately 1 nanometer thick. Now, when you extract current from the surface of a ferromagnet into an insulator, it turns out, because the material is magnetic, this means there are more electrons with their spins oriented in one direction than the opposing direction. This means automatically the current is spin polarized. Moreover, by choosing that insulating layer properly, the insulating layer itself, even though it's non-magnetic, can further spin filter the current, so that the current, which is tunneling across the insulating barrier from one layer to the next, can be up to more than 90% spin polarized. In other words, the electrical current consists 90% of electrons of majority spin polarization. The opposing magnetic layer, if it has the same magnetic orientation as the first one, can accept those electrons. There are empty states of the same spin polarization into which those electrons can tunnel. The tunneling process itself is ultra fast, and the spin orientation of the electron is preserved. However, if we change the orientation of that second moment, as shown in the bottom cartoon, so it's now in the blue direction, you can essentially switch off the current, because now there are no longer many states of the polarization of the tunneling electron. And so there are no states into which it can tunnel, and the tunneling current is reduced. Today, the difference in the tunneling current for these 2 orientations for interesting tunnel junction devices can be several times. So the tunneling magnetoresistance can exceed several 100%, even at room temperature. The metallic spin valve devices, even at room temperature, in the sandwich devices, the typical change in resistance is only 20%, but that still made them extremely useful to detect tiny magnetic bits. So the tunneling magnetoresisted device, using exactly the same spin engineering principles I discussed earlier, are today the devices that are used in magnetic disk drives to read information. But they also could form another type of memory, and this again is the proposal that I and some colleagues made in 1995, that we could use that same tunneling junction as a non-volatile element. The information is simply stored as the direction of magnetic moment. If it's red direction on the right, or if it's blue, one corresponds to a 0, another corresponds to a 1, and we can measure the state as a non-volatile state simply by the current, the magnitude of the current that is passed through the device. If it's a large current, then it's red. If it's a small current, it's blue. And we can do that very quickly on the scale of 1 nanosecond for typical devices. So in this cartoon, you can see these gray regions are the magnetic tunnel junctions, and they're interposed between two sets of copper lines in conventional CMOS technology. And now we can pass a current through a device from a word line through the bit line and detect whether the device sustains a large current or a small current, a 0 or a 1. And at the same time, we originally proposed we would simply pass currents along these word and bit lines to generate local magnetic fields to set the state of the magnetic tunnel junction. Today, we're going to use a totally different concept to change the magnetic state, to write the magnetic state by simply passing a greater current through the device, and that current carries spin angular momentum because the current is spin polarized, and that spin polarized current generates torques that can itself change the direction and set the state of these magnetic tunneling junction bits. Well, this concept of a magnetic random access memory, well, we proposed in 1995 when the devices, the tunneling junction devices at that time, had tiny tunneling magnetoresistance values of no more than 10%, and moreover, their resistance, typical resistance values were 100 million times too high to be useful. In order to reduce the resistance to useful values, the insulating layer has to be incredibly thin so that the electrons can readily tunnel across it. And that's something that we demonstrated a few years after we proposed this device. So on the one hand, these tunneling junction devices, properly spin engineered, can respond to tiny magnetic fields and can be used as the very sensitive detectors of magnetic bits and magnetic disk drives. And on the other hand, we could use them to build a very interesting non-volatile magnetic random access memory. Now, it took us several years to prove, not only find the materials, but to prove this concept. And I'm going to show you in the next chart the timescale for this. But eventually, we did build a few years ago, when I was at IBM, a 16 megabit fully functioning magnetic random access memory. And it has these attributes: extremely fast reading and writing, high performance, good endurance. Nevertheless, compared to the magnetic disk drives, it would be 100 times more expensive per bit. This is why magnetic disk drives are still useful today. But I wanted to just give you some idea of the time scale. Typically, from a material to an application, the National Academy of Sciences in the United States carried out a study a few years ago showing that it typically takes about 20 years, a long time from the discovery of a new material to its application. In this case, my discoveries in 1988, 1991 of these oscillatory interlayer coupling, the materials interface spin-event scattering, and the synthetic antiferromagnet, they were used in 1997 by IBM in the first spin valve recording head. It only took just a few years. And later on, 10 years later, the physics underlying the recording heads changed from a metallic system to the tunneling junction device, but with exactly the same, otherwise the same structures. Today, magnetic disk drives is a technology in some sense at the end of their roadmap. It's really very tough to further increase the capacity of magnetic disk drives for several reasons. So we need an alternative. Magnetic random access memory, you can see we proposed in 1995, and yet it was only 2 years ago that Samsung and later at TSMC announced it was possible to buy from them in their factories high performance foundry magnetic random access memory, because this memory has greater performance than, for example, embedded DRAM that it currently is replacing. So it's taken, as you can see, 25 years from the proposal to its application. This is more typical of complex materials. So nevertheless, magnetic random access memory, because it stores 1 bit essentially per transistor, nevertheless will never have the density of a magnetic disk drive, approximately 100 times different. So I proposed in 2002 third spintronic device. I called it a magnetic racetrack, which I think is super interesting. And over the last few years, we've demonstrated it's even better than when I proposed it in 2002. And the concept then is very different from all existing memory technologies and storage technologies today. As you can see in this schematic cartoon, the idea would be we would build a racetrack from a uniform magnetic material in the third dimension. And then, as you can see here, the blue and red regions correspond to different directions of magnetization perpendicular to this racetrack. But you can also see in the cartoon, what we do is we move the information which is encoded in the domain walls between the blue and red region. We move that information around the racetrack by passing current pulses into the racetrack. And these current pulses, if they're spin polarized, will develop spin torques onto the domain walls, which can move. My proposal was all the domain walls should move at the same speed together synchronously, so the information encoded as the presence or absence of domain walls can be brought to individual reading and writing devices built into the racetrack itself. In this way, because we would have 100 domain walls per racetrack, we would have 100 times the capacity, let's say, of a magnetic random access memory in the same size chip. This would enable us to have the capacity of a magnetic hard disk drive, but this device would be totally solid state without any moving parts and would be 1,000,000 times faster to access the first bit and would be totally reliable because there are no moving parts. So this is very interesting conceptually. And just wanted to show you when we first proposed this device, then as I said, it hadn't yet been demonstrated that a series of domain walls could be manipulated with the same current pulse. And this is something we showed a few years after I made this proposal. And in this cartoon, this is how it would work. Just as I said before, if you take current from a battery with equal numbers of spin up and spin down electrons in a magnetic material magnetized in the blue direction here, the current becomes spin polarized due to spin-dependent scattering. But because an electron with a spin has a spin angular momentum, and angular momentum is a conserved quantity, as you can see in the schematic cartoon below, in a racetrack formed from this orange material, the domain wall is shown here between two orange regions magnetized to the right and to the left. Then a conduction electron passing through this domain wall with a spin in a given direction, as it crosses the domain wall, it would adiabatically change its orientation as it follows the magnetization within the wall and would deliver a quantum of spin angular momentum to the domain wall. Because it's conserved, this would lead to the rotation of the direction of one magnetic moment, essentially one electron per magnetic moment. So with enough current of spin polarized electrons, we can cause the moments in the domain wall to rotate and the domain wall to move. And this is something that then we were first demonstrated in 2008. And in a paper we published in Science, where we showed in a wire we could create a series of domain walls. We could pass current pulses, move all the domain walls in the same direction, and prove the fundamental concept of this racetrack. We could current control the presence of a series of domain walls in nano wires. And therefore, this proved this concept I proposed 6 years earlier was valid. And if you like, this is the first edition of this magnetic racetrack memory. I don't really have time to discuss the evolution, but over the last 10 years, we've evolved this memory on racetrack, if you like, through 4 editions, 4 stages. And these are some of our own work, and of course, there's many papers from other groups around the world. But we moved from racetrack 1.0, in which the racetrack was formed from a very soft magnetic material, just as I've discussed, because in those soft magnetic materials, the current is strongly spin polarized. We then moved to, in racetrack 2.0, to a superlattice of cobalt and nickel, in which the broken symmetry causes the magnetization to be perpendicular and the domain walls to be 100 times narrower. Nevertheless, the domain walls move with current under the same principle that the current is spin polarized in the material and develops spin transfer torques onto the domain walls. In Racetrack 3.0, the Racetrack itself, again, is formed from a superlattice of ultra-thin cobalt and nickel layers, but it's deposited on this yellow layer, which is a heavy metal. And this uses a very interesting concept that when you pass current through a non-magnetic metal, which has strong spin-orbit coupling, that charge current will turn into a spin current that flows in a direction perpendicular to the charge current with a spin polarization, which is in a direction perpendicular to both the charge current and the spin current. So essentially, this converts a charge current into a pure spin current that carries spin angular momentum, and this can diffuse into the magnetic layer and apply torques onto the domain walls and cause them to move. And it turns out it can cause them to move much more efficiently than in Racetrack 1.0. And finally, we discovered just 5 years ago an entirely new Racetrack, which is based on this synthetic anti-ferromagnet that I first proposed 25 years ago for spin valves to eliminate the dipole fields. And it turns out that's also super important in Racetrack because the dipole fields emanating from one magnetic region to another will cause interactions, both within and between distinct racetracks. I want to briefly discuss that in a moment. So there are essentially 4 or 5 important ingredients to make this racetrack work. First of all, we make the racetrack from, as you see on the top right, these green and blue layers correspond to atomically thin layers of cobalt and nickel. Each layer is just a few angstroms thick, and the whole racetrack is only 1 nanometer thick. It's grown on a platinum underlayer, shown here as orange. And the platinum underlayer not only gives rise to this spin Hall effect, where current passed through that material generates a spin current, but it also gives rise to a very interesting exchange interaction, which is called a Dzyaloshinski-Maria exchange interaction that causes the magnetic moments in the magnetic layer on top of the platinum layer to want to be oriented perpendicular to one another. But it's a very special interaction in that it's a vector exchange, and that perpendicular orientation of the moments actually also has a chirality. So you can see in this cartoon, when we switch from a red region in the racetrack magnetized up to a blue region pointing down, the magnetization will rotate in a plane perpendicular to the domain wall, such that, as shown here in the cartoon by the green arrow, the magnetization in that domain wall will point to the right. But when in the same material, the magnetization rotates from a blue to a red direction, then the green arrow in the middle of the domain wall will point to the left. And the basic Dzyaloshinski-Maria interfacial interaction causes the domain walls in this perpendicularly magnetized cobalt nickel layer to both be what is called a nail domain wall and to have a chirality. The chirality can only be changed by changing that orange material. If we change it from platinum to tungsten, for example, the chirality will be different, but the chirality will be the same in all the domain walls. And this is the only reason why all those domain walls will then move in the same direction when we pass current pulses through that platinum layer, and which generates this spin Hall effect. Now, other elements of making racetrack very important are that the platinum typically will become itself slightly magnetic by a proximity-induced magnetization, and this synthetic antiferromagnetic concept is very important. So we will build the racetrack not from a single cobalt, nickel-cobalt superlattice, but from 2. I just want to remind you then about this Dzyaloshinski-Maria exchange, which also leads to chiral domain walls and more complex non-collinear spin textures, which, if there's time, I would like to discuss. On the left side here, you can see the basic exchange mechanism, which is called a Heisenberg exchange, causes in some materials the magnetic moments of the individual atoms, such as in cobalt, nickel, iron, to be oriented parallel to one another. And this is a Heisenberg exchange. In some materials, that exchange can be negative, and the moments will be oriented, not parallel, but antiparallel. This is an antiferromagnet, for example, nickel oxide. However, as I said, there are some materials where the symmetry is broken, no longer centrosymmetric, where with strong spin-orbit coupling, this leads to the magnetic moments wanting to be not parallel, as in a ferromagnet, or antiparallel, as in an antiferromagnet, but being perpendicular to one another in a helical or chiral fashion. And this then leads to, for example, as shown in the bottom, spin spiral states in some material, and if there's time to discuss, skyrmionic objects like this. But in the domain walls I've shown, it leads to the fact that they have a chiral nail structure. And this is shown here. I've just mentioned that. So this is the very important point, that the domain walls in this system are not only very narrow, they're only a few nanometers wide, but they also have this chiral structure. Now, the spin Hall effect I mentioned is shown schematically here. So, of course, you're all very familiar with a conventional Hall effect. You pass a current into a material in the presence of a magnetic field, and this will cause the electrons under Lorentz forces to be moved to the left or to the right, and this leads to the Hall voltage across the material. But it turns out in some materials, without any magnetic field and without any magnetization, you pass a yellow current here into, let's say, platinum, and you will generate a current of spin polarized electrons that flows in a clockwise or anti-clockwise direction around the current. And this spin polarized current can be extracted from the surface into a magnetic material to provide spin orbit torques. And it was only recently that it was realized that in metal systems, this spin Hall angle, the ratio of the magnitude of the spin current to that of the charge current, could be significant. Just 10 years ago, it was believed in metals that this spin Hall effect, the conversion of pure charge current to pure spin currents, was of the order of 1,000th, very, very tiny. But in the last few years, it's been demonstrated by ourselves and by others that the magnitude of these spin Hall angles, the efficiency of conversion of charge to spin current, can be extremely large, so that today we have materials where this efficiency is nearly 1. In other words, every electron that passes through a material like platinum or other materials will be converted to a pure current of a single spin polarized electron. And this is extremely important technologically. It turns out there's no time to discuss our work and others on extremely large spin Hall effects from non-magnetic triangular antiferromagnets and Kagome magnets, which are super interesting. Nevertheless, then, and I mentioned already that the neighboring domains lead to dipole fields, which cause interactions between magnetic bits in a racetrack. We have to eliminate them, and I mentioned earlier then this concept of a synthetic antiferromagnet, where an upper and lower racetrack, otherwise identical, one is a mirror image of the other through, in this case, I hope you can see, this ultra-thin ruthenium spacer layer that will give rise to a very strong anti-ferromagnetic coupling, where it turns out the chirality of these nail domain walls is preserved from one racetrack to the other. But in the end, you have a racetrack where there is no net magnetization anywhere in the racetrack, therefore no dipole fields. And nevertheless, when we did this, we discovered that current pulses passed across this domain wall would move it 5 times more efficiently than in an identical structure where the ruthenium thickness is changed by 2 angstroms and the coupling is ferromagnetic. So this is shown on the right-hand side when the magnetization in the upper and lower racetracks are exactly anti-parallel to one another and of the same magnitude. The efficiency of the motion, the velocity of these domain walls under the same current pulse intensity would be 5 times larger. And this led us to show that we could move domain walls in these systems at speeds exceeding 1 kilometer per second. And this leads to the possibility of creating devices that can operate on 100 picosecond timescales, which is faster than any conventional memory technology today. So this is very exciting. And this just shows you some images of racetracks in the laboratory. Now, these racetracks are too wide to be technologically useful. They're 2 microns wide. These are 50 microns long, but we can image them optically. The optical resolution is about 0.6 microns. Nevertheless, by applying current pulses, we can then image the structure of 20 domain walls, apply a current pulse, and we can see them move synchronously backwards and forwards, in this case, in a single racetrack at speeds of 300 meters per second. But in the bottom, you can see a synthetic anti-ferromagnetic racetrack where these domain walls are moving at 1 kilometer per second. So we're just showing you a series of images after we've applied ultra-short current pulses. And so basically, this is a very interesting racetrack memory technology where using synthetic antiferromagnetic racetracks, we can make technologically relevant devices. And we've demonstrated in my laboratory in Germany that we can shrink the width of these racetracks to technologically relevant sizes of the order of 15 nanometers. And it turns out the speed of the domain walls is unchanged for the same intensity of the current pulses. This makes this magnetic racetrack extremely useful. And I wanted to also mention that to build these devices, we built in Germany a series of deposition systems. On the left-hand side, you can see an orange long tube. These are heating tapes on an ultra-high vacuum tube, 15 meters long. And we can automatically transfer substrates on which we deposit these racetrack films between different types of deposition systems that are specifically designed to use different methods for depositing these ultra-thin layers. And again, there's no time to discuss this, but we have, for example, molecular beam epitaxy systems for depositing oxides and nitrides. We have a glove box for exfoliating 2D materials. And we have our own system, I call it Mango, which has up to 44 sputtering sources, magnetron and ion beam sources. And all these systems are designed so that the source materials can be rapidly changed while the rest of the system continues to operate. So this is a very exciting development that we've developed over the last few years to enable us to accelerate our discovery of interesting atomically engineered materials, both for spintronics and for superconducting applications and for other applications and science. Now, I don't know how much time there is left. I'm losing track of the time. Well, how much time do we have? Nobody knows, so I carry on. No. So I want to briefly discuss these magnetic skimions. And here I want to just turn to a very interesting family of compounds called Heusler compounds. Heusler compounds are a set of materials discovered several decades ago and were of interest because they involve no magnetic element, iron, cobalt, or nickel, rather manganese, and these materials were found to be ferromagnetic. They consist of 4 interpenetrating FCC face-centered cubic lattices. When the systems are magnetic, the lattice is distorted, becomes tetragonal, and this means that the magnetization will automatically be aligned along a particular direction, the tetragonal direction. And it turns out that these systems display very interesting spin textures, and I want to briefly discuss one of these. So these, we have time?
Yes.
Okay, so then the structures I want to discuss are called skimions and antiskimions. So maybe some of you are familiar with this. It's a relatively recent discovery, which is that in a material like this Heusler compound, where the moments want to be parallel along a particular direction, it turns out that under some temperature and field regimes, instead, the magnetization will break up into an array of skimionic objects. And the reason is the same Dielinski-Maria exchange interaction that favors perpendicularly magnetized moments. This means that you can lower the energy of the system by forming, as shown in this cartoon, circular objects in which there is a domain wall in which the moments are aligned perpendicular to one another. So the block skimion has a structure in which if you go from the outside, where the moments are out of the plane, to the inside of the object, where the moments are in the opposite direction, the moments will rotate from the up to the down direction in a plane which is tangential to the surface of this object. It's called a block skimion. The important point is that rotation of the magnetization will either be clockwise or anti-clockwise, depending upon the material system. Alternatively, one can form a nail skimion, as on the right-hand side, where the magnetization rotates from an up direction to a down direction in a plane along the radius. This is a nail skimion, and basically if we just looked along one radius, that would be exactly like the chiral domain walls in the racetrack that I briefly discussed. In both the block skirmion and the nail skirmion, these are typically found in cubic systems, so that the circumference, the structure around the outer edge of these objects would be identical in all directions. However, it was theoretically predicted that in some systems with lower symmetry, you would, instead of seeing these two types of block and nail skirmions, you could find what is called an antiskirmion. And in an antiskirmion, the structure of the boundary between the outer material magnetized up and the inner material magnetized down changes around the circumference between nail and block-like segments. And every time you go from a nail to a block-like segment, the chirality changes. And this object was theoretically predicted, but had never been observed until we first observed it 4 years ago. And I want to show you how we observed it. We observed it using Lorentz transmission electron microscopy. So we basically take a single crystal of this material. We use focused ion beam milling techniques to cut a sliver, a lamella, out of this material, which is sufficiently thin that the electron beam can be transmitted through it. And that means it has to be, roughly speaking, below 200 nanometers thick. So we make this using focused ion beam milling. When the electrons then, high energy electrons, 300,000 volt electrons, are passed through this lamella, they will be subject to Lorentz forces from the magnetic component of the magnetization in the plane of the film. And this means that if we have a block skirmion, then in the Lorentz microscope, this will look like this, as you can see here, this white blob of increased electron intensity. On the other hand, the nail skirmion is invisible because the Lorentz forces cause the electrons to be rotated around the circumference of the object. So there's no change in the intensity. On the other hand, the antiskirmion we calculated should show an image in the Lorentz microscope of these two white dots of increased intensity and two black dots of decreased electron intensity. And we saw this, and I hope you can see in the top column to the left in part A, you can see such an object. This is, this is obtained by passing a very high energy electron beam through an ultra thin lamella and detecting the electrons that are subject to the Lorentz forces in that lamella. Now it turns out you can also see in E, F, G, H, and the rest, you can see an array of these skirmions. And this is typically what you find. You find an hexagonal array of these skirmionic, anti-skirmionic objects. Well, this was the first time that these had been observed. That was just 4 years ago. We're very proud of this achievement. And I wanted then briefly to discuss, turns out these anti-skirmions have super interesting properties. Firstly, if we take a series of Lorentz images in different magnetic fields, if you look at the row of images shown in B, on the left-hand side, you can see nothing. And that's because if we apply big enough magnetic field, we will see all the magnetic moments actually become parallel again. And the Lorentz imaging technique is only sensitive to changes in magnetization, not in magnetization. But as we reduce the field, you can see in these row B, you'll start to see, going from left to right, the anti-skirmions appearing, until on the very right-hand side, you can actually see a different chiral spin texture, which is a helical spin texture, which is found in zero magnetic field. So in this case, the array of anti-skirmions has disappeared, and instead the system energy is stabilized by forming helical structures. And it turns out in these anti-skirmion systems, the helical structure is directed along one of two perpendicular in-plane directions. And it turns out this helical structure has very interesting properties. Maybe you can see here in this Lorentz micrograph on the right-hand side, you can see a series of white and black lines. These correspond to the in-plane component of the magnetization, changing from a right to a left direction. And you can see as we go from the right to the left in this lamella, the thickness of the lamina was designed to increase. And you can see that the spacing of these white lines increases. And it turns out the period of this helix increases monotonically with thickness. And now if we switch to a different technique, we can use magnetic force microscopy to image these anti-skirmions and helical structures in thicker lamella. We found that indeed the period of this oscillatory variation in magnetization increases monotonically with thickness, here up to 1.6 microns, and here even up to 4 microns. And you can see beautiful images of this helical structure in the magnetic force microscope. We're sensitive to the outer plane component. And if we change the magnetic field, you can see these white dots appearing, which are the anti-skirmion objects. And the size of these objects also depends on the lamellar thickness. And without going into any details, this is because of long-range magnetic dipole fields that depend on the thickness of the lamella. This had previously not been seen. Now, just to, turns out these spin textures, why skirmions and anti-skirmions are interesting is that the spin textures are topological. That means it's very difficult to distort them into any other structure. But we found in this system a new type of topological spin texture, these elliptical block skirmions, who's oriented along two distinct crystal directions. Unfortunately, I'm running out of time, and I also want then, so I'm going to skip this part. Turns out by changing temperature and field, we can change these objects. And I just wanted to briefly mention that recently we've discovered an even more interesting object, fractional antiskirmions at the edges of these lamella. Hadn't been theoretically predicted, but we believe this is a consequence of the interior structure through a bulk boundary correspondence. And these fractional antiskirmions only appear at the edges, and this is being confirmed theoretically by our collaborators at the University of Basel in the group of Daniel Loss. This work is not yet published. And so I'm going to skip this. This concept of two distinct topological spin textures in the same material could lead to racetracks, where one topological spin texture, like an antiskirmion, could represent 1, and the elliptical block skirmion could represent zeros. And I think I have no time to talk about our work in 2D materials. I should summarize. So I want to summarize by saying that racetrack memory, where we store information in the presence or absence of chiral domain walls, has become even more exciting in just the last few years, as we've developed and discovered in synthetic antiferromagnets new mechanisms that allow for these chiral domain walls to be moved 10 times more efficiently than was previously thought possible. And if we've already demonstrated we can make racetracks that are just 50 nanometers wide with the same high efficiency of motion of the domain walls. This means we could build racetrack devices that could replace magnetic disk drives with super high capacities. And at the same time, if we build a racetrack which is super short with a single domain wall, we could replace the fastest memory today, static random access memory, because this single domain wall racetrack can be ultra fast, 100 picoseconds. We've already demonstrated in the laboratory below 400 picoseconds, would use much lower energy, would be non-volatile, and would have substantially increased density compared to today's static random access memory. In the last part of my talk, I briefly discussed the recent discoveries of ourselves of antiskirmions and elliptical block skirmions, these topological spin textures, and we've seen a whole variety of chiral spin textures in a range of compounds that I didn't have time to discuss, including 2D materials that today are of great interest. And I want to thank you very much for your attention. And here I wanted just to briefly acknowledge some of the people involved in the work I've discussed, including my students and postdocs at the Max Planck Institute for Microstructure Physics. Some of the samples I discussed were prepared by Claudia Felser and her collaborators in Dresden. And I also wanted to acknowledge our theoretical collaborations with Daniel Loss and his collaborators and Ingrid Mertig at the Martin Luther University. Thank you very much for your attention.
Yeah, thank you, Professor Parkin, for the very nice talk. I'd like to invite him to join me over here as we have a chat and try to answer questions from the floor.
Okay.
Right? Yeah. Thanks, Stuart, for the very nice recount. Especially enjoy your personal recount of how your ideas and spins and inventions in spintronics led us from Gutenberg all the way to the cloud. And perhaps now to the metaverse, right? And
definitely.
Thanks for the nice tutorial there as well. Show us all the spin physics behind, and more exciting is the exhilarating ride that you brought us through in the last part on racetrack memory, skirmions, anti-skirmions. Yeah, I'm sure.
Thank you very much.
I'm sure our audience, are looking forward to more from you. They have questions here, and
Okay.
quite a few of them.
Okay.
Maybe I'll begin with the first one. So Neil Kundu asks, can the quantum effects of electron spin, example superposition, entanglement, be used to further improve memory storage capacity similar to quantum computing? What are your thoughts?
Okay, so I think that question is, of course, spin is quantum mechanical, and there are ideas about using spin qubits for quantum computing, which I think are very, very interesting. It's not something that we're doing ourselves. Actually, what we're doing is imagining ways of building cryogenic memories using concepts from spintronics plus exotic superconductors, which we can build at interfaces between conventional superconductors and topological metals.
Right, thanks. Harutai has this question. Thank you for the great ideas and for your sharing. He asks, could you please advise what are the advantages of using Racetrack memory compared to 3D NAND memories that we have now?
Okay, so the advantages are actually NAND is a relatively slow technology to access the bits. So Racetrack would enable us to write and read much faster than is possible in 3D NAND. Also, 3D NAND is a fantastic technology in which, in order to avoid the problems with, as you basically, it's a capacitor which stores a certain number of electrons. But as you shrink the dimensions, you cannot store enough electrons to enable multi-bit storage. And this is how you get to the very high storage capacities of 3D NAND. So Samsung invented a fantastic technique where they built layers of these memory cells and could access them all vertically. However, that technology relied on the flash memory cells, each cell being relatively large, and relies on many, many layers. Well, this technology only works up to 100 layers or so. And this means that it's impossible, it's very difficult to increase the capacity further. You need to do something else. Magnetic Racetrack memory, if you could build the vertical one, this would be super great. Very tough to build three-dimensional structures, although there's a lot of exciting research in that area in magnetic 3D systems, including our own work. But you could actually build Racetrack again, one on top of each other. And it turns out you don't need many Racetrack layers to compete with the highest capacity 3D NAND, but they would be much faster and would actually be more reliable because you rely on these magnetic elements. There's a huge numbers of advantages, mostly speed compared to 3D NAND.
Oh, that's exciting to hear. Thanks. Jay has a question. I've encountered magnons when studying roto-model-like Hamiltonians. Can skyrmion physics be studied using a similar model? What are some fundamental differences between these two?
So I'm really not entirely sure what you mean, but nevertheless, it turns out that the properties of spin waves or magnons in chiral systems are extremely interesting compared to conventional systems. And in principle, because of the topological nature of these textures, you could imagine that the lifetime of these excitations would be longer. And this leads to some very interesting consequences.
Right, thank you. I think we have come to our final question already. But perhaps I have a personal question before we finish off, and that would be what in your mind would be the timeline towards seeing some of these Racetrack or skyrmion type memories come to fruition?
Oh, that's a very tough question to answer. But of course, we hope that it will be sooner rather than later. And I think it really, in the end, depends on convincing the mass manufacturers like Samsung, TSMC, to make the investments that will be needed to demonstrate that you could reliably make billions of these Racetracks. And I think that's the fundamental question. A lot of it then is driven by existing memory technologies that they're more familiar with. But as you find it's not possible to further increase the capacity of conventionally, you have to find something else if you really want to build, let's say, digital data storage systems with greater capacities. So it's really a question of how soon will these existing technologies no longer be improvable? And that's probably right now. So then it means that within a relatively short space of time, you will need alternative technologies that have superior performances or the potential to go beyond. And I think this is what has happened, like MRAM. It was, it took a long time, but partly because existing technologies could be evolved. And since one is more familiar with them, you're more confident that you could evolve them. But today, I think with the ending of scaling of many, many, many technologies, it means you have to do something new. And this huge opportunity then for spintronic devices, which can scale to tiny dimensions or give you much greater performance with greater reliability. So huge opportunities. And I think this is the time when we will move from, completely charge-based to many more spin-based technologies, both for memory and even potentially for computing.
Right. Really, thank you,
thank you,
Stuart, for this very inspirational talk. And we really enjoy this. And thank you, too, for bringing Spintronic to the mess. Really.
Okay, thank you very much.
And we really look forward to some of these new ideas that you have coming to fruition.
Okay, excellent.
Thanks, everyone, for joining us.
Yeah, thank you very much.
Hello, my name is Andrew Hawley, and I'm an assistant professor in the National University of Singapore's Biomedical Engineering Department and a PI at the Mechanobiology Institute here in Singapore. I'm very honored to be introducing our next three speakers, all of whom are young scientists in the process of making important contributions to a wide variety of biomedical fields. Our first speaker is Elam Yaini, who is a PhD student in Professor Ronit Sachi-Finaro's lab at Tel Aviv University. Elam will be sharing work on understanding the complex relationship between glioblastoma cancer cells and their microenvironment in the brain.
Hello, everybody. My name is Elam Yaini, and I'm a postdoctoral fellow at Ronit Sachi-Finaro's laboratory at Tel Aviv University, Israel. I would like to thank the organizers for the opportunity to present here today my PhD project, which focused on the role of P-selectin in glioblastoma progression. So glioblastoma, or in short GB, is the most common and lethal type of brain cancer with a median survival of 20 months. The available treatments for these patients mainly include surgical resection, followed by chemotherapy and radiotherapy, and other targeted therapies or current immunotherapies have showed limited efficacy in glioblastoma patients. Nonetheless, it is known that the brain microenvironment facilitates glioblastoma progression. So we focused on microglia, which are the brain-resident immune cells. They have antigen presentation, cytotoxic, and phagocytic abilities. However, they can be found in several activation states, which can be roughly divided into the pro-inflammatory antitumorigenic phenotype, in which they can attack cancer cells and recruit other immune cells to the tumor, or the anti-inflammatory protumorigenic phenotype, in which they possess angiogenesis and immunosuppressive characteristics and facilitate tumor progression. The mechanisms which induce this phenotype in glioblastoma are yet to be fully understood. So to investigate the interactions between glioblastoma and microglia cells, we first established several human and murine glioblastoma mouse models and found activated microglia in the tumor site in all the different models and in patient FFPE samples using IBA1 immunostaining. We then proceeded to in vitro experiments where we co-cultured labeled glioblastoma cells with unlabeled microglia cells and followed for cancer cell proliferation. We found that as more microglia in the culture, the higher was the proliferation rate of glioblastoma cells. To evaluate the effect of microglia on glioblastoma cell migration, we have used transfer migration assay where we see the microglia cells in the lower chamber and glioblastoma cells in the upper chamber. And we found microglia to significantly enhance glioblastoma cell migration. Now, to elucidate factors that are involved in these interactions, we have again co-cultured glioblastoma cells and microglia cells and screened for over-secreted factors following these interactions. Using both patient-derived and murine glioblastoma cells, we found high secretion of P-selectin in the co-culture medium compared to the monocultures. P-selectin, or in short CELB, is an adhesion molecule involved in leukocyte rolling, and P-selectin and its primary ligand, PSGL1, have been shown to be involved in several immunological processes, as well as in cancer spreading and immunity. So exploring the expression of P-selectin in glioblastoma, we found that glioblastoma cells not only secrete P-selectin, but also express P-selectin on their membrane, and that both secretion and expression were elevated when glioblastoma cells were treated with microglia condition medium. Moreover, we found microglia cells to express high levels of PSGL1 when treated with glioblastoma condition medium. These results suggest that P-selectin expressed on glioblastoma cells may interact with PSGL1 expressed by microglia cells. So we went back to our mouse models and found high of P-selectin and PSGL1 in tumor areas enriched with activated microglia. To validate the relevance of these findings, we have collected FFPE samples from short-term survivors and long-term survivors glioblastoma patients and found much higher expression of P-selectin in the short-term survivor samples compared to the long-term survivors or healthy human brain samples. And these findings were further validated using the TCGA database. So we decided to continue and evaluate the functional role of P-selectin in glioblastoma progression. To that end, we have generated P-selectin knockdown glioblastoma cells using shRNA, and we established a glioblastoma 3D model using a drop method, in which we create multicellular spheroids, see them in matrigel, and follow their invasion. We observed an increased invasion of glioblastoma cells when microglia were incorporated into the spheroids, and reduced invasion following P-selectin knockdown. To validate that these effects are due to P-selectin knockdown, we have used P-selectin small molecule inhibitor and found similar results. We then asked what is the role of this axis in microglia immunophenotype. So we've treated human microglia with a recombinant P-selectin and found increased expression of the immunosuppressive cytokines IL-10 and TGF-beta, a reduction in the phagocytosis ability, and reduced nitric oxide release by human microglia. Interestingly, we could reverse this phenotype by blocking P-selectin or PSGL1, but not by blocking the other P-selectin ligands, CD44 and CD24. This indicates that P-selectin promotes the immunosuppressive phenotype of microglia by specifically binding PSGL1. So we proceed to in vivo experiments, and we inoculated intracranially into mice the P-selectin knockdown glioblastoma cells and found reduced tumor growth and prolonged survival in the P-selectin knockdown group using patient-derived and murine glioblastoma mouse models. Immunostaining of these models revealed reduced proliferation, increased apoptosis, reduction in blood vessel density, and increased infiltration of cytotoxic T cells and reduced infiltration of T regulatory cells. These findings regarding the T cells were also validated using flow cytometry. So to further characterize the changes in glioblastoma tumors following P-selectin knockdown, performed single-cell RNA sequencing, which allowed us to have a closer look into the different cell type populations and subpopulations inside the tumor. Looking at the cancer cell populations, we found a reduced expression of the invasion, proliferation, and angiogenesis signatures in the P-selectin knockdown tumors, and in the microglia populations, we found an increased expression of the pro-inflammatory antigen presentation and T-cell recruitment signatures, which correlates with our previous observations. Finally, we evaluated the therapeutic potential of P-selectin inhibition by treating glioblastoma-bearing mice with the P-selectin inhibitor IV, and we found reduced tumor growth in human, murine, and genetically engineered glioblastoma mouse models. Now, these findings, together with the observation of improved immune activation, have led us to try and combine P-selectin inhibition with other immunomodulators in order to harness both the microglia and the T cells against the tumor. So we have synthesized an anti-glioblastoma nanovaccine composed of PLGA nanoparticles loaded with glioblastoma neoantigens. The nanoparticles were decorated with mannose residues in order to target dendritic cells, which will then activate T cells to specifically recognize and attack the glioblastoma cells. We have treated glioblastoma-bearing mice with a P-selectin inhibitor IV every other day, and with 3 doses of the nanovaccine once a week intranasally. While the nanovaccine by itself did not affect much tumor growth, the combined treatment has showed synergistic effects in reducing tumor growth and prolonging mice survival. So this is what we are working on now, as well as other combinations with other immunomodulators. So to conclude, we've showed that P-selectin mediates glioblastoma cell migration and proliferation, that P-selectin and PSGL1 axis promotes the pro-tumorigenic phenotype of microglia. We demonstrated that P-selectin inhibition reduces the tumorigenic potential of glioblastoma cells in 2D and 3D culture models and leads to delayed tumor growth, prolonged survival, and improved immune infiltration in vivo. These findings were validated using several human and murine glioblastoma models, which represent the different glioblastoma subtypes. And for the future, we are now planning to conduct clinical trials evaluating the anti-cancer effect of an FDA-approved anti-P-selectin antibody, which is currently indicated for sickle cell disease. So I would like to thank all the lab members, our collaborators, and of course, my supervisor, Professor Ronit Sachi-Fainaro, and thank you for listening.
Okay, thank you very much, Elam, for this really nice talk. As always, please feel free to drop your questions into the Q&A. I guess I can start us off with a question. I think it's really fascinating how you're looking at these, trying to untangle the contributions from these two distinct components of the glioblastoma, right? So one of the interesting aspects of glioblastoma is the stiffening, right, of the tissue surrounding this tumor. And I'm wondering if in your SELP knockdown tumors, if you have measured or maybe noticed any change in the matrix mechanics surrounding that tumor?
Yes, so actually we didn't check these, but what we saw that after treating with a P-selectin inhibitor or P-selectin knockdown, so the tumors are more encapsulated, so which is basically a good thing because glioblastoma is also known to be a very invasive tumor. And then let's say that after surgery, it will be more possible to resect the whole tumor or most of the tumor after following these treatments. But it's interesting what you're saying. We can also examine this.
Excellent. I mean, one of the things I'm also wondering is you showed this increased sensitization to the chemo drug or the chemo therapeutic treatment. What do you think is driving this?
Yeah, so microglia are also known to increase drug resistance in glioblastoma, and actually we are working more on combinations with other immunomodulators, vaccines or immune checkpoint blockade. But we also believe that, and we also see that it improves also the activity of chemotherapeutics, but we are still evaluating these mechanisms deeper.
Cool. Yeah. So we do have a question from the audience from Nishama Galad. It says, very interesting work. Does the approved P-selectin blocker pass the blood-brain barrier?
Yeah, so this is a very good question. We don't know. And for our research in mice, we used small molecule inhibitors, hydrophobic molecule, which did pass the blood-brain barrier. And the reason we want to use this antibody, it's for the sake of the patient, actually, because this is already approved and in use for other indications. So we think it's worth the try. And one more aspect is that we also observed similar effects on bone marrow-derived macrophages. So we believe this can maybe also affect these cells. But yes, this is one of our major concerns using this antibody, and we hope we are now conducting preclinical investigations with this antibody, so we'll have more data about it.
Okay, I guess one other thing I'm kind of interested in is, do you think this is specific to a glioblastoma kind of cancer model, or do you think some of these similar pathways might be playing a role in other tumor types?
Yeah, so actually we did observe a high expression of P-selectin in melanoma and breast brain metastasis, and we have also some preliminary results using melanoma 3D models. And some of the experiments that we performed were using a system with only microglia or macrophages, and we saw the influence of P-selectin, PSGL1 axis on their phenotype. So yes, we believe there is also it is also relevant for other tumors or other even immunodegenerative diseases. So yeah.
Okay, so is this the plan next, to try to administer a P-selectin knockout or interfere with SELP for breast cancer or colon cancer, something like this? Or are you focused more on sort of secondary metastases? from an initial
Yes.
brain cancer?
Yes, so now we are focusing on glioblastoma and melanoma brain metastasis. But yes, this is one of our plans to also examine other primary tumors and see the role of B-selectin and their interactions with immune cells.
Okay, excellent. Thanks again for your nice work. Oh, actually, we have one more question that just came in. Katie Croft's wondering, have you considered a combination with therapies, such as focused ultrasound with microbubbles to alleviate this blood-brain barrier issue, which might allow temporary permeability?
Yeah, this is very interesting. We actually started a collaboration with another lab in our institute that is specialized in this ultrasound BBB opening. And so, yes, we are looking into it. Unfortunately, I don't have any data at the moment, but we really hope it will improve the antibody penetration.
Excellent. We'll look forward to seeing that data next year, maybe. Okay,
Yeah.
thanks, Elam. I think we can move on to the next talk at this point. Our next speaker is Andrea Lupi, a PhD student in clinical neurosciences, working in Professor David Manone's lab at the University of Cambridge. Andrea's presentation today will highlight the relationship between different regions in the brain during neural information processing.
Hello, hi, and welcome, and thank you for your interest in my work. So I'm Andrea Lupi from the University of Cambridge, and my PhD is about understanding how our species got from here to here. And of course, that is thanks to the human brain. Now, to understand the way that we think about the human brain, it's helpful for us to go together through a little murder mystery. Imagine that you are a detective trying to figure out which of these individuals committed a particular murder. Now, as a detective, you may have found a witness who tells you that the culprit belonged to the suit of hearts. This lets you eliminate some of the potential suspects. And if in addition, your witness also tells you that your culprit was a man, you can identify some further suspects to eliminate. But still, you don't uniquely know the identity of the culprit. However, you may also have a second witness who also tells you that the culprit belonged to the suit of hearts, but adds the information that they wore a crown. Once again, on its own, this information would only let you remove some of the suspects, but not all of them. Crucially, however, if you put these pieces of information together, you can now uniquely identify the culprit as the King of Hearts. Now, the reason why this is important is because you can tell that there are different kinds of information that were being provided by the different witnesses. Some of this information, like the belonging to the suit of hearts, was redundant because each witness provided that information, meaning that you would have had access to it, even if one of them had forgotten about it. Some of the other information was uniquely provided by one or the other witness. But most importantly, the information about the identity of the culprit was only available when the information provided by the two separate witnesses were combined together. That is, there was a synergy between these two witness testimonies. This is important because we now have the mathematical tools to quantify in a formal way synergy and redundancy in information provided by different sources. And this is possible over the last 10 years, thanks to the tool called information decomposition. And my work is about trying to identify within the human brain where is synergy most prominent. And in particular, to do this, we used functional MRI data obtained from human participants and made publicly available as part of the Human Connectome project. And this, using the tools from information decomposition that we just went through together, allowed us to identify in the human brain where synergy and redundancy are most prominent. And in particular, as you can see here on the bottom right, the distribution of synergy shown in red and of redundancy shown in blue is not random. And using a meta-analysis of several thousand published studies, what we were able to find is that they also have different associations with different kinds of tasks, such that redundancy is most associated with sensory and motor low-level cognition, whereas synergy is especially associated with high-level cognition. And this association is not just within the human brain, but rather it also holds across different brains. So we compared humans and macaques with data from functional MRI, and what we were able to find is that the two species do not differ in terms of the proportion of total information that is explained by redundancy. On the other hand, we also found that humans are significantly more rich in synergistic information compared to macaques. And the evolutionary relevance of synergy does not stop there, because we were able to find that synergy is especially prominent in regions that have expanded the most over the course of evolution, and it is also especially prominent in regions of the human brain that are especially enriched for uniquely human-accelerated genes. Now, we wanted to go a bit deeper and find what are the underlying neurobiological underpinnings of synergy. And what we found is that synergy is especially prominently associated with synapses, which are the locations, the physical locations that enable neurons to communicate and integrate information with each other, which is what synergy is all about. And we got there through two convergent lines of research. First, we carried out a data-driven analysis of gene expression patterns. And what we were able to find using these post-mortem gene expression data is that regions that are especially synergistic are the same regions that are especially enriched for synapses and synaptic transmission. And we confirmed these results using in vivo synaptic density mapping through positron emission tomography, which showed once again that the more synergistic a region is, the more it expresses synaptic density. So just to summarize our research, what we found is that neural synergy supports higher cognition in the human brain. And within the human brain, it is also especially associated with evolutionary expansion and especially associated with the expression of evolution-related genes. In addition, we find that synergy is especially enriched in the human brain compared to the brain of macaques, which is not the case for the other main kind of information, that is redundancy. And finally, looking at the neurobiological underpinnings, we were able to find that through the divergent and convergent means, synapses are the macroscale and microscale underpinnings of synergy in the brain. And overall, we think that this is prominent and promising in terms of establishing a way to identify what makes the human brain so special. Of course, this was a synergistic effort, and I wish to thank everybody who contributed to this, both people and institutions. And in particular, I wish to express my gratitude for my funders, Gates Cambridge, and also to my supervisor, Dr. Stamatakis from the University of Cambridge. And finally, here is how you can contact me, and I look forward to answering your questions. Thank you for your attention.
Andrea, thank you very much for this deeply synergistic work, as you kind of said. So as always, please feel free to leave questions for anyone who's watching who might want to ask Andrea something into the chat. But again, I guess I'll start it off. So this isn't my field, but I'm very fascinated by this idea that you can look across different organisms to see, actually, this is where some of our higher order functioning as humans can kind of come in just by looking at different regions in the brain and how they're overlapping. How much is this conserved to some extent as we move further back in the evolutionary tree? Like if you looked at mice or something like this, would you find results like this, a continuation of this trend where there's less density in these overlap regions?
I think that's a great question, and I just want to say it's a real pleasure to have the opportunity to discuss this research with the other attendees. So going to your question, that's something that we're actually interested in delving deeper into. Right now, we were lucky to have the ability to compare humans with a relatively similar species, macaques, who we can scan in a similar kind of environment. Of course, the situation becomes more challenging as the species diverge more, because then it's not clear how much of the differences can be attributed just to differences in information capacity and how much instead depends on other anatomical aspects. But it is exactly the right kind of question that we're also very interested in.
So then I guess I might continue this, and I know it's not trivial to find model organisms, so I'm certainly not saying go out and find these organisms to do it on, but in other kind of kingdoms of life where you have evolution of increased intelligence, if you were to compare, say, fish and dolphins, do you think you would also see a similar increase in that particular, those particular characteristics you saw on the primate chain?
I hadn't actually thought about the comparison with marine life. I think, once again, the differences between fish and dolphins might not be just in terms of brain size and cognitive capacities, because, of course, dolphins are also mammals. So there is also that additional part of the differences that might need to come into play. But we do think that there is something that has to do with, in general, what synergy enables any information processing system to do. And to the extent that we can refine that from a theoretical standpoint, then I think we will be in a better experimental situation to be able to ask exactly what kind of question we can predict that would be related to synergy, and obtain an understanding that is independent of the implementation, if you will, and just really depends on the capacity of the system itself.
Sure. So the synergy really might be sort of universal as we talk about an increase in intelligence. That's fascinating. So Neshama Gilad says, great talk. What are the plasticity rates of synergy? Are there any changes with age, for example?
That's also something that we are looking into right now. So the data that we have come from the Human Connectome project, which is a data set of relatively young and healthy adults. But for instance, we have some follow-up work that is looking at what happens in patients who have suffered a disorder of consciousness. So they've, for instance, in a vegetative state as a result of brain injury. And in that case, what we see is that there are changes that involve synergy as well. So that is not the same as answering the question of what happens with aging, but I have colleagues, some of my co-authors on this paper, in fact, who are also looking at gradual changes in a very large database called CAMCAN that comprises ages from 18 to 80. And there are changes that occur both in terms of synergy and in terms of redundancy. So part of the question now is, how do we interpret them? What do they mean? But there do seem to be changes of this kind, yes.
Okay, great. We also have a question from Anonymous, who says, do we know what could interfere with or improve such synergistic neural interactions?
So something that we also know, in addition to brain damage, which of course happens to have a lot of different changes on brain function, but we have also compared that with the brain changes that happen when someone undergoes anesthesia, which is something that happens millions of times every year in the world for, of course, surgery. And luckily, it is reversible. People are anesthetized and something changes in their brain, but then as the drug wanes, its effects also wane and people come back. And we have seen that these effects are relatively similar to what someone who is in a vegetative state will have. But the difference being that in the case of anesthesia, this is a transient change and then everything goes back to normal. So that tells us that synergy can be perturbed pharmacologically, but that this perturbation will also be reversed once the pharmacological intervention is wanes off.
So I kind of want to follow up a little bit on this. Do you think it's possible for us to train the synergy in our brains? If you tracked a single person, as they do the crossword puzzle every single day for 2 months or something, would you see an increase in that density, more synergy in the brain? Or do you think that each one of us have our own kind of levels?
That's a fascinating hypothesis. I don't have evidence right now to say it's for or against it. But I do think it would be interesting because, of course, you mentioned what happens with aging, but something that we're also very interested in is how synergy develops into adulthood and throughout, from fetus to newborn to then into adulthood. Does that change with education? And that's something that we currently don't know, but I think it would be very interesting to find the answer to. And likewise for artificial systems. We currently don't know whether different ways of training an artificial system might be especially suitable to induce synergy there with its advantages and all of that. Yeah.
Excellent. So we have a lot to look forward to. So thank you very much, Andrea. And now I think we'll move on to our final talk of this session.
Thanks.
Our final speaker is Sherry Ann Ng, a research associate in bioinformatics in the Department of Metabolism, Digestion, and Reproduction at Imperial College London. Sherry-Ann received a PhD in biomedical science at Murdoch University in Perth, Australia, studying sepsis in preterm infants. Today, we'll hear about new work on specific maternal factors that play a role in preterm birth risk and sepsis.
Thank you to the organizers for letting me present my research, which is on insights into preterm birth using multi-omics approaches. So why is preterm birth important? Preterm birth affects 15 million babies every year, accounts for more than 1 million deaths globally, and is a substantial economic burden on the healthcare system. Preterm birth is very complex to solve. There are a range of factors that can cause preterm birth, from smoking to ascending infections, or the cause could be unknown. Babies who are born preterm are also at higher risk of developing infections as well as other long-term morbidities. So how can we understand preterm birth better? Increasing studies show the potential of multi-omics approaches to capture the complexity associated with preterm birth. There are a range of omics fields, from genomics to metabolomics. These can give us different levels of insight into how different biological systems are contributing to the phenotype, which is the physical trait we see happening. Today, I will share two of my research projects, which focuses on understanding preterm birth from a microbiological perspective in both the mother and the baby. The first uses multi-omics approaches to improve our understanding of bacterial bloodstream infections, also known as neonatal sepsis, in preterm infants. Neonatal sepsis accounts for 15% of deaths within the first month of life, causing significant mortality and morbidity, especially in preterm infants. There are serious implications due to the lack of early detection, prompt diagnosis, and appropriate treatment of sepsis, as well as limited understanding of early life immune metabolic responses. Our study combined the use of both transcriptomics and metabolomics to identify blood biomarkers that is distinctly present during sepsis in preterm infants born less than 30 weeks gestation. Following patient recruitment and sampling, we processed whole blood for transcriptomic RNA-seq analysis, metabolomic liquid chromatography mass spectrometry analysis, and integrated different data types for patient classification. Based on overall gene expression, the transcriptome of septic infants, the ones in red, appeared to be more similar to each other, but different from infants with possible or no sepsis, the ones in green and blue. More importantly, the blood leukocyte transcriptional responses of septic infants was significantly altered compared to infants with no sepsis, with 1,317 differentially expressed genes. Based on network analysis and pathway overrepresentation analyses, we identified that these transcriptional aberrations in host responses during confirmed sepsis were associated with four main functions. First, that pathogen and pattern recognition mainly function through toll-like receptor pathways, which is not surprising, considering these receptors recognize a range of bacterial components from both gram-positive and gram-negative bacteria that are often implicated during sepsis. Second, for cytokine signaling, we found that both pro-inflammatory cytokine signaling and immune inhibitory signaling were linked to upregulated genes. These were mainly your interferon and interleukin signaling genes, as well as immune inhibitory signaling genes such as IL-1R2 and IL-1RN, which inhibits interleukin-1 signaling. Third, that various pathways associated with immune and hematological regulation were overrepresented. More importantly, that despite most genes being upregulated, two key genes important for regulating apoptosis, BCL2 and IL-7R, were downregulated, suggesting that genes involved in apoptosis are likely dysregulated during confirmed sepsis. So the importance of crosstalk between metabolism and the immune system for host responses during infection is increasingly being recognized. Here we found connections between immune and metabolic genes that are known to drive inflammatory responses. We also found overrepresentation of pathways linked to cholesterol metabolism, suggesting that altered cholesterol mechanisms could exacerbate inflammatory responses in infants with confirmed sepsis. In our metabolomics analyses, we identified 36 metabolic features that were potentially important for separation of infants with confirmed sepsis and no sepsis. And more specifically, based on these metabolic features, majority of infants with sepsis had similar metabolic profiles to each other that were distinct from infants with no sepsis, as can be seen from the heat map. Additionally, we also putatively identified 5 with biologically relevant metabolite features, including fatty acids and pyruvate, which have been shown to be increased in septic patients in existing studies of immunometabolism during sepsis. So does including multi-omic data improve patient classification? Yes, so when we included demographic data and laboratory data only, we generated 3 clusters. All possible and no sepsis infants were in a single cluster. However, majority of sepsis infants were in one of 2 clusters. When we included transcriptomic and metabolomic data, we had 6 clusters. Again, all infants with possible and no sepsis were in a single cluster. However, now each infant with sepsis were in individual clusters. Of course, this was only a pilot study, but we postulate that integration of multidimensional demographic, laboratory, and multi-omic data has potential applications for stratified medicine. This is a figure from one of my publications, and the main takeaway is that we are currently in this era of empirical medicine, where we are using a one-size-fits-all approach to sepsis diagnosis. However, when we start to include omics data, we could potentially stratify patients into sepsis clinical groups. Eventually, when we include continuous real-time clinical measurements, we could potentially move towards precision medicine, where we personalize the diagnosis and treatment for these septic babies. Ideally, sometime in the future, we would move towards preventive medicine, where we can have predictive biomarkers for sepsis, or we can prevent babies from even being born preterm, since they are most at risk of developing neonatal sepsis. And this leads me to another aspect of my research work, which focuses on the maternal side of the preterm birth equation, where we have used other omics approaches to improve our understanding of the relationship between the vaginal microbiota and preterm birth. So increasingly, the role of microbes, such as in the oral and gut cavities or in the lower reproductive tract, they have been recognized as important in shaping health and pregnancy outcomes. But what do we know about the vaginal microbiota and preterm birth risks? So a lactobacillus dominant vaginal microbiome is characteristic of healthy pregnancy, and specifically, lactobacillus crispatus has largely shown to be beneficial, whereas iners and high diversity type microbiomes have been linked to higher rates of preterm birth. Also, during pregnancy, bacterial vaginosis, or BV for short, is linked to a 2-fold increased risk of preterm birth. Geographical and ethnic differences can also influence the relationship between the vaginal microbiota and preterm birth risk, and that this association is different in Black and white women. Now, despite the fact that most preterm births occur in Asia, majority of existing studies that have looked at the relationship between these factors and preterm birth risk have mostly focused on North American and European populations. Our study was a large-scale characterization of the pregnancy vaginal microbiome and cellulase activity in a low-risk Chinese population. So the main takeaway is that we collected vaginal swabs from 2,796 pregnant Chinese women and performed 16S rRNA sequencing. We then classified 2,689 samples that passed initial criteria into vaginal microbiome groups that were based on the most dominant species in the vaginal microbiome, as shown in the table. We also conducted a sub-cohort analysis in 819 women who had available data on cellulase activity, which is often used as a marker for microbiome dysbiosis in China, and leukocyte wet mount results, which has been used as an indicator for inflammation. As expected, majority of Chinese women in our study were characterized by lactobacillus dominans, and mostly lactobacillus crispatus and iners dominans. The proportion of crispatus and iners dominans in our population were very similar to Chinese and Korean women, so these are the pie charts with the green circles. When compared to non-African Americans and Caucasian women in Europe, the pie charts with the yellow circles, in those populations, the ratio between crispatus and iners dominated microbiomes tended to be larger, with majority being dominated by crispatus. On the contrary, when we look at Asian and Black women in Europe and women in India, as well as African Americans, we find that in these populations, there tended to be larger proportions of iners dominated microbiomes instead of crispatus, as seen by the red circled pie charts, or they had overall low levels of lactobacillus, as seen by the black circled pie chart. So our findings suggest that ethnic and or geographical differences are a major source of variation in the underlying structure and composition of the vaginal microbiome. In our sub-cohort analyses, we found that high cellulase activity was associated with high diversity vaginal microbiomes that were dominated by BV type taxa, and specifically taxa such as Gardnerella and Atopobium, which is characteristic of microbiome dysbiosis. When we compared preterm and term births, we found that microbiome dysbiosis, high cellulase activity, and leukocyte presence were not significant drivers of preterm birth in this cohort of Chinese women. So generally, the risk of preterm birth in our cohort was around 5.4%. In the overall population in China, it's around 7%, which is still lower compared to the global average of 10%. So our findings suggest that the lower risk of preterm birth may be due to differences in the vaginal microbiota and or host immune responses of Chinese women. Finally, I would like to reiterate the potential of multi-omics approaches to capture the complexity associated with preterm birth, where we have the possibility to generate new insights that help us to predict and prevent preterm birth, as well as improve the health outcomes for these babies born too early. I would like to acknowledge the participants of our studies, as well as all the people, institutions, and organizations that have made this research possible. Thank you.
Sherry-Ann, great work. I think this talk and this paper that you described really illustrates the potential for this omics data and how it can impact us in our daily lives, right? How we can help people around us. So while we wait for some questions to come in, I kind of have a bit of a basic question, I suppose. What's the next step? So it seems like you can identify mothers that might be at risk prior to birth. So does this mean that how these mothers are actually treated during the delivery is changed, or what happens at this point?
Yeah, so there are many, actually, I talked about how there are many causes to preterm birth. So at least for my research, we can look at introducing lactobacillus probiotics to the mothers when they are having their pregnancy, and this could probably prevent them from getting preterm birth. So there are trials currently ongoing, and our group does have studies in that area. So I think that's something that we could potentially apply in the future to try and help mothers and reduce the risk of preterm birth.
Okay, so it's not just about addressing the sepsis that comes with the preterm birth, but actually preventing it itself. Is that correct?
Yeah, so preventing the mom, so basically looking at the mom's vaginal microbiome and trying to make it better by introducing more probiotic type bacteria, and that would result in the mom having less risk of developing infections and reduce the risk of also preterm birth. So then if the babies come out at term, then they themselves have less risk of developing sepsis. So it's sort of like a transitionary effect.
So I guess what I'm wondering is this the only way of kind of preventing that sepsis, by trying to push it to a full-term birth? Or are there other interventions where maybe you can't prevent the preterm birth, but you can address the sepsis that is so often associated with it?
Yeah, so I think it's very difficult because a lot of the times when the babies come out, they have very immature immune systems. So until we can find a way to expedite the maturity of the immune systems, it's very difficult to prevent the sepsis because you need the host response to be mature enough to overcome the infections. So I think in that sense, we are still not there yet, but hopefully one day we could see how we can maybe expedite the maturity of the immune system, or at least prime it to be better at targeting infections.
Yes, hopefully. We have a question from Philip Carlson at Uppsala University. They say, thank you for a very interesting presentation. Do you know how much food choices might affect the vaginal microbiota?
Yeah, actually, I'm not sure what's the answer to that question because I haven't actually looked at the nutritional side of things and how that affects the microbiome. But I think potentially it could affect the microbiome, but I can't say I know the answer to that for sure. But thank you for your question.
So, okay, one more question from Martin Toole. Thank you for the talk. Based on the microbiome analysis and your results, is there anything one can do to minimize the risk of preterm birth in addition to avoiding typical bad habits like smoking?
Yeah, so there are a lot of things that contribute to the risk of preterm birth. I mean, if a mom is overweight, so if they have higher BMI, then that also contributes to higher risk of preterm birth, higher maternal age as well. And if mothers have gestational diabetes, all these factors do affect the risk of preterm birth. So apart from just smoking, there are a lot of other factors that you can try to reduce or prevent yourself from getting in order to increase your chance of having a term delivery.
Interesting. One other really fascinating thing I think I saw on one of the slides is that in the future plans, it's maybe not just testing once to get this this omics data, but maybe serial collection of data throughout the pregnancy. So I'm wondering if you've kind of tried this already, and if you have, if there's ever a time where you see that things are going well, and then quickly goes bad, or vice versa, where you have all these risk factors and then suddenly something changes and they're no longer at the same level of risk.
Yeah, so our group has done a lot of studies on, has done a few longitudinal studies where they have tracked the microbiome across different stages of the pregnancy. And they do see that if, let's say, there is a drop of crispatus, then there's a higher risk of the mom having more diversity. And when they have more diversity, it leads to a higher risk of preterm birth. So in that sense, yes, we've done studies on that, and we continue to do studies on that as well.
Cool. We have a question from Leo Chan who says, thank you for the talk. May I ask if symptoms or even resolution of inflammation through breast milk feeding is considered passive immunity, if this is critical for development?
Yeah, so I'm actually not sure how to answer that question because I'm not really in the breast milk field. But I would assume that breast milk would be extremely important for development, and it would help to prime the immune system, especially in preterm babies. So obviously we know that breast milk does pass on antibodies. So this would be very helpful for preterm babies as well, to give them some sort of boost to their immune system.
Okay, we have a question from Javaria Zafar who says, thanks for the talk. Will there be tailored treatments for every woman, since there seems to be varying vaginal microbiomes, at least across international borders, let's say?
Yeah, so I think that would be the ideal place that we can move to. So I think I said in my talk that we hopefully can move towards preventive medicine or even precision medicine. So this is where we start to tailor the treatments and the diagnosis for women or even babies with sepsis. But I don't think we are there yet because there's so much variability and so much that we still don't understand in terms of, host microbiome interactions. So until we understand better, it's very hard to tailor the treatment for each women. Yeah.
And our final question is from Anonymous, who says, more generally, what are some of the known factors or risk factors that influence vaginal biota?
Yeah, so ethnicity does influence the vaginal microbiota. We can see that women with diabetes may also get different microbiotas. So there are a number of factors that can actually impact on whether the mom gets a bad microbiota or not.
Okay, so thank you again, Sherry-Ann, for this very interesting talk. And thank you to all of the speakers for participating today in this session. Thank you to all of you for watching and participating so nicely with questions and observing the talks. And we begin again tomorrow at 8 a.m. Singapore time for registered participants. Thank you very much.
Thank you.